Good morning, everybody. Respected speakers, faculty, students, and audience. Welcome to the third and final day of NWNTD 2020, sixth workshop on nanoelectronics and theranostics in post-COVID-19 healthcare. The forenoon session for today includes lab demonstration and online webinars by academic experts. So without further ado, let us commence today's events. In the first video, Mr. Lakshmi Raman Adil will present the comprehensive making of spilled VTM kit. He is a PhD scholar in the Department of Chemistry and pursuing research under Professor P.K. Iyer. Spilled VTM kit is specially designed by IIT Guwahati and RR Animal Healthcare Limited to collect and transport virus samples to the testing center by maintaining the viability and the virulence of the sample for successful detection of COVID-19 virus. I request Mr. Adil to proceed with the presentation.
Thank you, Konika. Very good morning to all. Today I will demonstrate how to prepare SPLD BTM kit. But before that, I would like to talk about the background of our work. The first human COVID-19 case was reported in Wuhan city, China in December 2019. But reason of this disease and how it is spreading was unknown. But that time we thought that this place Wuhan is 1,000 kilometers far away from our place, so we cannot affect by this disease. On January 14, WHO confirms coronavirus is spreading through human-to-human transmission. And on January 30, the first case was found in Kerala, India. In February, there is no significant rise of cases, but in month of the March, transmission of virus grew up very rapidly beyond any control. That's why government was forced to prevent the spreading of coronavirus. They imposed nationwide lockdown and they have imported the COVID-19 kits for perform large amount of testing. But these corona kits are not sufficient for us because we have large population. Then government is looking on premium institutes for prepare COVID-19 test with the slogan Atmanirbhar Bharat, Atmanirbhar Assam. My supervisor, Professor Iyer sir and Professor Ghosh sir have taken this as a challenge to develop COVID-19 kits. Because of their constant guidance, support and their leadership, we can able to develop the COVID-19 in collaboration with RR Animal Healthcare. So in my part, I am going to demonstrate how to prepare SPILD VTM. Please have a look on our tutorial video. Corona pandemic situation, it is extremely important to perform large amount of testing so that we can identify a person who is infected with the SARS-CoV-2 virus. Presently, we don't have any vaccine. Many countries, including India, trying very hard to development a vaccine. But before that, a large amount of testing is the only option so that we can prevent the spreading of the coronavirus. And for successfully diagnosis of the coronavirus, the quality of the specimen is very important. In collection purpose, we want some transporting media, that is VTM. VTM is viral transport medium. It prevents sample from the contamination and it keeps viral agent safe for longer time. For preparation of VTM, we need glasswares, pipettes, and chemicals. To maintain the sterility of the medium, we have autoclaved all the glasswares and milky water. Also, we will be using laminar aeroflow hood to make VTM. Now, I am going to show how to prepare the SPLD VTM. We will start with 5 liters of autoclave milky water. Then we will add chemical components according to a standard protocol. You can see we are adding all chemicals very carefully, and then we will shake reagent bottle very vigorously to make solution homogeneous. Then we will add antifungal and antibacterial agent to inhibit any growth of unwanted agents. And after the addition, we will properly mix it. And then we will add phenol red. Phenol red is a pH indicator and it changes color if there is any growth of contaminants. At the end, we are filtering this media by using 0.2 micron filter membrane to make it sterile. Prepare our media, and we will distribute 3 ml VTM into each vial. The SPILD VTM kit qualifies all standard criteria mentioned for viral sample collection. The nasal and oral swabs provided in the kit are flexible to avoid discomfort to the patient. The swab also has a break point so that the swab can be broken after sample collection. Now our SPLD testing kit is ready. Transport to the testing center.
After sample collection, RNA isolation is one of the important steps during COVID-19 test. RR Animal Healthcare Limited and IIT Guwahati has developed the RNA isolation kits. In this video, Dr. Anil Bitkar will talk about the development of RNA isolation kit, and he will also perform the RNA isolation using these kits.
Hello everyone, I'm Anil. I'm a PhD from Professor S.S. Ghosa's lab at Department of Biosciences and Bioengineering, IIT Guwahati. Currently, I'm working as a scientist at RR Animal Healthcare Limited. In this series of video lectures, I will talk about the basic principle and the working of viral RNA isolation kit and RT-PCR based COVID-19 detection kit. As we know that the SARS-CoV-2 or the COVID-19 virus is a RNA virus, and for the detection of this virus particle with RT-PCR based technology, we need to isolate RNA from these samples, and thus we will be using viral RNA isolation kit developed by IIT Guwahati and RR Animal Healthcare Limited. Once the sample is collected in VTM, this sample will undergo RNA isolation. The RNA purification kit has been developed with guanidinium isothiocyanate based lysis method and silica gel membrane based purification technology. This kit provides highly pure RNA from oral, nasal, or the nasopharyngeal samples. As we know that in COVID testing, we have to isolate the RNA from both the human cells as well as virus particles. We have optimized this RNA isolation kit for human cells and virus particles. During the RNA extraction process, the RNA virus containing samples will be lysed by lysis buffer, which is the highly concentrated solution of guanidinium isothiocyanate. As the SARS-CoV-2 virus is a RNA virus, we have avoided use of proteinase solution, and this resulted in the isolation of highly pure RNA from both the human cells and viral samples. The addition of ethanol in the RNA containing sample makes RNA ready for binding to the column, and simultaneously the addition of RNA carrier into the sample improves the viral RNA yield. Now let's start with the RNA isolation process. For your protection, always wear lab coat, gloves, safety glasses, mask. We have to clean our work area, prepare our wash buffers and the lysis buffer, and gather all the ingredients or the materials needed for RNA isolation process. Now let's start with the RNA isolation process. For SARS-CoV-2 testing or COVID-19 testing, the samples should be collected at hospital or the testing center. The first step involves use of lysis buffer for lysis of cells as well as virus particles. So we will take around 150 microliter of sample and we will add a to the sample. I am transferring 150 microliter of viral transport media containing sample to these 1.5 ml microcentrifuge tubes. Now I will add 600 microliter of lysis buffer to the samples. This lysis buffer contains carrier RNA molecules which helps to increase the RNA yield. After the addition of lysis buffer, we have to mix this sample thoroughly, and for the mixing purpose we can use pipetting or vertexing. To ensure efficient lysis, it is essential that the sample is mixed thoroughly with the lysis buffer to yield a homogeneous solution. After vertexing the samples, we have to incubate the samples at room temperature for 10 minutes. For optimal lysis of viral particles, 10 minutes room temperature is required. In this incubation stage, most of the potentially infectious agents or the virus particles are inactivated in this. Now we have to add 600 microliter of ethanol to each sample. Addition of ethanol, we have to mix these samples with the pipetting. Now, after the addition of ethanol, the RNA is ready to be captured on spin column or the silica column. So, in the next step, we will add around 700 microliter of sample to the silica or the spin column. Now, we will load these samples to the silica columns. So, these are the silica columns, and we have to number or name these silica columns according to our sample numbers. Now we have to transfer the RNA containing sample to the silica columns. Now after the addition of RNA containing sample into the silica column, we will close the lid and centrifuge these columns at 10,000 rpm for 1 minute. After the centrifugation step, we can see that most of the sample has been filtered through the spin column. Now we will throw this filtrate and add the remaining lysate to the column. We have to again centrifuge these columns at 12,000 rpm for 1 minute. Now most of the RNA has been captured on the silica column, and in the next step we have to wash the debris or the contaminants which are attached on the column. For this we are using different wash buffers like wash buffer 1 and wash buffer 2. For the washing steps, we are transferring these columns to the new collection tubes. I am transferring these columns to these new collection tubes. So now we will add 500 microliter of wash buffer 1. So now we will add 500 microliter of wash buffer 1 to each column. After the addition of wash buffer 1, we have to centrifuge these columns at 12,000 rpm for 1 minute. For next wash buffer step, we have to again transfer these columns to new collection tubes. After, after transferring these columns to new collection tube, we have to add 500 microliter of wash buffer 2, and again we have to centrifuge these columns at 12,000 rpm for 1 minute. After this second washing step with wash buffer 2, the RNA is ready to be eluted. But before that, we have to make sure that no wash buffer 2 is present in the column, and for that we will transfer these columns to new collection tube and will spin out these columns at maximum speed for 1 minute. After the dry spin of this column for 1 minute, we have removed the extra wash 2 buffer present on the column, and now we can elute the RNA from these columns. For the elution of RNA, we have to transfer these columns to the new collection tube, and we'll add around 50 microliter of elution buffer to these columns. For RNA elution, I am adding 50 microliter of pre-warmed elution buffer. After addition of the elution buffer, we have to store or incubate this column at room temperature for 1 minute. In the next step, we have to centrifuge this column at 10,000 rpm for 1 minute for RNA elution. After centrifugation step, the RNA will be eluted in the tube. This RNA can be stored at -20 or -70 degrees Celsius for further use. For RT-PCR reaction, 2 to 5 microliter of RNA can be used as a template.
RT-PCR is considered the gold standard test for COVID-19. Again, RR Animal Healthcare Limited and IIT Guwahati have also developed RT-PCR kits. In the next video, Dr. Anil Bidkar will explain the principle behind the development of these kits while performing the experiment itself.
My name is Anil and I'm going to explain you a complete standard operating procedure for COVID-19 detection real-time PCR kit. Initially, I will explain the basic principle behind the design of COVID-19 detection kit, and in the next step, I will perform the real-time PCR to explain you the complete procedure and the data interpretation. This kit has been designed on the principle of multiplex PCR. Multiplex PCR is a widespread molecular biology technique for amplification of multiple targets in single PCR experiment. In a multiplexing assay, more than one target sequence can be amplified by using multiple primer pairs in a single reaction mixture. Before we start with the RT-PCR, we need to know what is the PCR or the polymerase chain reaction. So I'll give you a short introduction about the PCR or the polymerase chain reaction.
Polymerase chain reaction, or PCR, is a technology indispensable to genetic and molecular biology. Many of its applications are widely used in the world around us, including DNA sequencing, DNA fingerprinting, forensics, detection of microorganisms, and diagnosis of hereditary disease. PCR allows fast and inexpensive amplification of DNA fragments. Because it is able to generate large quantities of DNA from a small amount of nucleic acid, PCR is also referred to as molecular photocopying. PCR depends on a series of 20 to 40 repeated cycles of DNA replication by a DNA polymerase enzyme. After each cycle, the number of DNA strands is doubled, and at the end of a 40-cycle reaction, more than 1 trillion copies are generated from a single copy of a DNA molecule. The PCR process is separated into 4 steps: initialization, denaturation, annealing, and elongation. In the first initialization step, the reaction is heated to 94 to 96 degrees Celsius for 2 to 10 minutes to activate the DNA polymerase and to denature other contaminants in the mixture. In the case of colony PCR screening, this step also facilitates cell lysis to release DNA and denature other cellular proteins. In the denaturation step, hydrogen bonds between the double-stranded DNA are broken by heating the reaction to 94 to 98 degrees Celsius for 20 to 30 seconds. After the DNA strands are separated, primers bind to a complementary sequence in the DNA template to guide DNA polymerase replication. In this step, the reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds to allow for optimal primer annealing. After the primers establish a starting point for the enzyme, DNA polymerase starts to incorporate deoxynucleotide triphosphates in a 5' to 3' direction to synthesize a new DNA strand. The temperature and extension time for this elongation step depends on the type of DNA polymerase enzyme and the target amplicon. Commonly used Taq polymerase polymerizes at a speed of 1 to 1.5 kilobases per minute and works ideally at 72 to 78 degrees Celsius. The cycle then repeats from denaturation to elongation 20 to 40 times. At the end of the last cycle, there is a final elongation step that keeps the reaction mixture at 72 to 78 degrees Celsius for 5 to 15 minutes. This ensures that any remaining single-stranded DNA is fully extended after the last PCR cycle. A final holding step keeps the PCR at 4 to 15 degrees Celsius for an indefinite time, keeping the products for short-term storage.
To understand how amplified PCR products, also called amplicons, are detected in real time, let's first review the events that occur during a normal cycle of the PCR reaction. Recall that the first step in any PCR cycle is to raise the reaction temperature and melt double-stranded DNA. Then, when the temperature is lowered, the specific primers bind to the sequences at each end of the target DNA. The intervening DNA can then be synthesized by polymerase reaction in opposite directions. As a result, you produce two double-strand copies of the target DNA when you started with only one. If you have any confusion about this basic process, it might be a good idea to review the program on basic PCR once again. To detect the generation of new amplicons in real time, the PCR reaction requires an additional ingredient, a single-stranded DNA probe designed to hybridize to the part of the DNA sequence synthesized between the two primers. However, unlike the primers, this probe is modified in a special way. One of its nucleotides is labeled covalently with a fluorescent molecule, and another nucleotide is labeled with a fluorescence quenching molecule. The quencher rapidly absorbs any light energy emitted by the fluorescence molecule as long as it remains in close proximity. Now, let's look at what happens when this additional ingredient is present during a single cycle of PCR. As the primers bind to the separated strands of DNA, the probe also finds its complementary sites between them. The enzyme that synthesizes new DNA from the ends of the primers also has a second activity, an exonuclease activity. So when it encounters double-stranded DNA in its path, it will disassemble the strand that is in its way and replace all of the nucleotides. As the polymerase cuts through the probe, note that the nucleotide bearing the fluorescent marker and the one bearing the quencher are separated from one another. In the absence of a nearby quencher, the fluorescent molecule can now emit detectable light when stimulated. Each time another amplicon is produced, another fluorescent marker is released from its neighboring quencher. Therefore, just as the number of amplicons doubles in each PCR cycle, the amount of emitted fluorescent energy also doubles. This light generation can be monitored during the PCR reaction in a thermocycler that is equipped with a fluorometer. So, if you begin with a clinical sample that had only one copy of the target DNA, it could take 40 or more cycles before the amplicons are detected by a fluorometer in a specialized thermocycler. However, if the original sample contained 32 times more copies of the target DNA, then the fluorometric detection would occur after 5 fewer rounds of PCR. And if there were 1,024 more target DNA sequences in the original sample, then the fluorescence signal will be detected 10 rounds earlier. So, the amount of specific DNA in the clinical sample is determined by reference to the round of PCR in which the amount of fluorescence first crosses the threshold of detection.
Now,
RT
in case of single reaction mixture. Now, in case of our COVID-19 detection kit, we are going to detect 3 different genes. Out of these 3 genes, 2 genes are from viral origin and the third gene will be detected from human cells. As we know that detection of every gene needs a pair of primers and a detection probe in the reaction mixture. Thus, in COVID-19 detection kit, to detect 3 different genes, we have a mixture of 3 pairs of primers and 3 fluorescent probes. Now I will talk about the genes which we will detect using this PCR. The first two genes, that is N gene and RDRP gene of virus origin, will be detected to confirm the presence of coronavirus. Now the N gene is also known as a nucleocapsid protein gene, which packages the virus positive strand, that is RNA, into a helical ribonucleocapsid, which plays a fundamental role in the replication of virus. And the probe that we will be using to detect the N gene contains a FAM fluorophore, which gives green fluorescence. The second gene that we are detecting from virus is a RDRP gene, which codes for RNA dependent RNA polymerase. The RDRP, or the RNA dependent RNA polymerase, replicates the virus genetic material inside the cells. Thus, the probe detecting the RDRP sequence of virus is conjugated with ROX dye, which gives orange fluorescence. The third gene that will be detected or amplified is RNSP, also known as RP, from human cells. As we are collecting the samples from nasal or oropharyngeal region, along with the virus samples, we are also collecting human cells, and thus the samples will show the presence of RNSP gene or RP gene. The probe, which is specific to RNSP or the RP gene, has hex dye and it gives yellow fluorescence. Now let's start with the RT-PCR reaction setup and the experiment. These are the components of our kit. The first one is 2x RT-qPCR master mix. This master mix contains all the nucleotides and reverse transcriptase as well as polymerase enzyme. The second component is a reference dye. The third component is COVID-19 primers and probes. This is a mixture of all the three primers, pairs, and probes for three different genes. The next component of this kit is a positive extraction control, which contains the RNA isolated from a COVID-19 positive person. Following this, we have negative extraction control, which contains the RNA isolated from healthy or COVID-19 negative person. Now I will start with the RT-PCR setup. Initially, we have to take PCR tubes and label them according to the sample name. For today's experiment, I am taking tubes for positive extraction control, negative extraction control, and the negative control or the water control. So here I am taking the required tubes and labeling them according to the sample number. Once we are done with the labeling the tubes, we have to thaw our primer probes and master mix and other components. Now our kit components are thawed and we have to mix the reactions according to the protocol given in the kit. So this is the protocol that we will be giving with the kit, and we have to add these reaction components. We have to set up the reactions according to the protocol given in the kit. We have to mix these reaction components according to the table given in the kit manual. We can either distribute these reaction components into each tube individually, or we can make master mix of all the components except the template of positive extraction control or negative extraction control as well as water control. So here I am preparing the master mix for all the tubes of positive extraction control, negative extraction control and water control. I will distribute these I will distribute this reaction mixture into different tubes, and in the end I will add the template of positive extraction control RNA, negative extraction control and the water control. So I'm adding all the reaction components into one tube and I will distribute it into different tubes. Now I have distributed the reaction mixture into different tubes of positive extraction control, negative extraction control and the water control. Now I am adding the template of positive extraction control, negative extraction control and the water control. After addition of the templates or positive extraction, negative extraction and water control, we have to mix these reaction mixtures. The reaction mixture in the tube can be mixed with pipetting or by vertexing. Here I am using vertexing for mixing purpose. We have to do a brief vertexing for 2 to 3 seconds for each tube. After vertexing, we have to spin down these tubes to bring all the volume of reaction mixture at the bottom of the tubes. Now, after spin down, we will directly put our tubes into RT-PCR instrument. The RT-PCR instrument contains different slots for different tubes, which contains the specific number for each slot. So we have to remember or note down these slot numbers, and in the end we can name our samples according to the slot number.
look at the when you need something like a PowerPoint, then the live pause is a really good function to use. So previously in vMix you're able to use the pause function on video files, and now you can use it on live sources. So I'm going to show you how to set that up now. So in vMix you'll see here that we have
For artificial reaction, we have to set up the reaction temperatures according to the table that is given in the kit manual. Now, the first step is a CDNA synthesis, and the later these three steps are for artificial reaction. As we are detecting three different genes, we are selecting three different detectors to collect the fluorescence from the dyes which are attached to three different probes of the genes. Now I have started the reaction and will wait for reaction to end, and then I will start with the result interpretation. For result interpretation, we have included this table in the kit manual. According to the interpretation table, when we see a signal or the amplification of N gene and RDRP, we can say that there is a presence of viral RNA in the sample. Additionally, if we are collecting the sample from human patients, we will see the amplification of RP gene. And when we can see the amplification of all the three different genes, we can say that the sample is COVID-19 positive. But in case of negative persons, we will see the amplification of only RP gene. This plot shows the fluorescence in FAM channel or the detector containing FAM signal. So here we are seeing the amplification of N gene. We can see that there is amplification of N gene in case of positive extraction control. This plot shows the fluorescence signal for ROX dye, which is for RDRP gene from viral origin. Now, here also we can see that the amplification of positive extraction control sample, which shows that there is presence of RDRP gene and N gene in case of positive extraction control sample. The third plot here shows the hex fluorescence for RP or the RNase gene, and here we can see that there is significant increase in the signal of RP gene amplification in case of negative extraction control. We can also see that there is slight increase in the fluorescence for positive extraction control sample.
From these plots, we can say that the positive extraction control sample shows the amplification of N gene, RDRP gene as well as RP gene, which confirms that the sample is a positive. And in case of negative extraction control, we are seeing that there is no fluorescence in N gene amplification plot or the RDRP amplification plot. We are only seeing the amplification of RP gene or the hex dye channel, which confirm that this is a negative extraction control sample.
Hi, my name is Anil and I'm going to explain you a complete standard operating.
Thank you, Dr. Anil and Mr. Adil. We will now be taking questions from the participants. So our first question is, if a person is infected with common cold or some other virus which is similar to coronavirus like SARS or MERS, how will this detection kit differentiate?
So, Konika, this is Professor Ghosh.
Yes, sir. Yes, sir. You're audible.
Yes. So basically, Anil probably could not able to connect. The answer to the question is these primers and the kit is specific for COVID-19 only. So other SARS viruses cannot be detected with this kit. So this is specific for the COVID-19. As you have all seen, the primer, the probe for the N gene, RDP genes and everything that is specific for the COVID-19. So other viruses will not be detected. This is specific.
Okay, sir. Thank you, sir. Our next question is, these days many false positive or false negative results are also seen in detection kits of corona. Why is it so and how can it be controlled?
The answer to the question is these again depend upon the probes. If you talk about the RAT kit, that is the rapid antigen kit, which is the first kit. One second. So the RAT kit is not very specific kit, but the RT-PCR kit is supposed to be specific. But there is a WHO and CDC guideline. Based on that, the primers and the probes are made. Now, few of the genes that we have seen from our experience are very specific in terms of detection. But if you take some other genes, then they may vary little bit. So a different company sells different kind of kits. So and all the kits have more than one probes and primers, and it depends upon the CT value, depends upon the RNA quality of RNA isolated in the process. So all this adds to the factor, and whatever in 33 or 33 CT value, below that it is thought to be positive, but sometimes because of the poor RNA quality and because of sometimes the primers are not very good in terms of their the fluorophore which is attached, sometimes detached. So those gives a little bit of background signal and false positivity. But there are few kits which are gold standard, like in vitrogen kits, our kits, and many other people are scientists have used across the board, and they are found to be specific. But there are some kits actually which gives false signal. So there are many kits which have been certified by this time by ICMR, but eventually they are not found to be consistent in terms of the reproducibility, batch to batch, RNA extraction, and the gene specificity, all this matters. So that a small fraction becomes false positive. So those are the cases. Thank you.
So Konika, I also want to add something, because we are not prepared for this corona pandemic, and due to lack of the training of the technician and huge workload on them, that's why we are getting this thing.
Konika. Hello, Konika.
Anil, you are
audible.
Yes.
Yes, sir.
Yes, sir. In order to avoid these false.
1 second, Anil. If you are here, I will not intervene. You take care of this. Okay. Thank you.
Yes. To avoid the false positive and false negative samples, that's why we have included. We are detecting 3 genes. So after including 3 genes, we can say if there is a false positive for positive samples, there should be a signal for all the 3 genes, and in case of negative samples, there will be a signal for one gene, that is RP or RNSP. If there is problem, or if there is any issue during the sample collection or RNA isolation, we may not get the presence of RNSP, and there we can detect whether this sample is not showing false negative. And in case of say false positive also, we will get around 2 genes of viral origin, but we may not get the RNSP. So in that case, we can identify whether this is false positive or false negative. The false positive cases are coming because of the non-specificity of the primers and the probes, or there may be a chance of degradation of the probes due to long-term storage at room temperature or at higher temperatures. So basically we have to take care of all the things, keeping the primers probes at the cold conditions, and while thawing we have to take care that those probes should be kept at on ice or at cold temperatures. And I think that is the answer of this question.
Anil, in addition to that, her question was probably directed to the other kids. Why in other places, in some instances, false positive cases are being found? So you have already given the answer. So the reason may be it is the non-specificity of the probe and not proper way of handling. So those are the cases. So that is why some kits are very reproducible and being used in practice by all clinics and clinicians.
Yes.
Thank you.
Thank you, Professor Ghosh sir and Dr. Anil. Another question that we have is, how long the virus sample remain viable in the VTM media?
Well, after sample collecting, the viruses will be viable for a very long term, because in VTM we are providing most of the contents which are already present in the serum, and there are many instances where they are finding that the viruses are alive, and the people who are working on RNA isolation, or whoever is handling those samples in laboratory for testing purpose, those people are also getting infected with those samples. Uh, Obviously, they will be taking care of all the essentials which are in the lab. That means they will be working with PPE kit and mask and all the gloves and other things. But still, those samples or those viruses have been found as alive, and they can infect the people.
even after say 48 or 72 hours of sample collection. That's why we this buffer, which is called lysis buffer, that actually inactivates or lyses the virus completely, and it has been seen that the virus will or the virus particles will become ineffective after the lysis, but it can be stay infective as long as it is in the VTM. So those people who are working in the lab, they have to take care of that thing.
Thank you, Dr. Anil. I think that clarifies the question. Last question that we have is, what is the use of specifically 0.2 micron filter during the preparation of the VTM media?
Yes, during the VTM preparation, we are adding lots of chemicals, or say we are also using FBS, and we have seen that many protein aggregates are also present in the FBS, and sometimes uh, there might be a chance of bacteria which could interfere, which can go in the VTM preparation, and if we do not filter those bacteria out, essentially it will grow after storage and it will destroy the media. We won't be able to use the VTM after say for 10 hours or 24 hours of the preparation. So we have to filter out all the debris of proteins, aggregates, bacteria, whatever is there in the VTM. You have to filter all the things, and essentially after filtering out through point 2, we will make the VTM as a sterile VTM. We will be adding some antibiotics also, but still we have to make it clear so that it can be used properly in the RNA isolation or during the sample collection. Does that answer?
Yes, thank you, Dr. Anil. Yes, that answers the question. Thank you, everybody. So now we will break for half an hour and then move on to our next session.
Thank you.
Welcome back, everybody. So our next speaker is Professor Aditya D. Mohite from Rice University, USA. Professor Mohite is an associate professor in the Department of Chemical and Biomolecular Engineering and directs an energy and optoelectronic devices lab, working on understanding structure, function properties, and materials, with the aim of controlling charge and energy flow across. His research philosophy is applying creative and out-of-the-box approaches to solve fundamental scientific bottlenecks and utilize the same to demonstrate technologically relevant performance in devices that is on par or exceeds the current state-of-the-art devices. He has published 150 peer-reviewed papers in journals such as Science, Nature, Nature Materials, Nature Nanotechnology, Nanolitters, ACS Nano, Chemical Society Reviews, Applied Physics, Letters, and Advanced Materials, amongst others. He has also delivered more than 90 invited talks. The title of his talk is Semiconductor Physics of 2D Inorganic Perovskites. I request Professor Mohite to proceed with the talk. Please, sir.
Thank you very much for the invitation. Just give me a second. Okay, let me see if you can see my screen. Can you see my screen?
Yes, sir, we are able to see your screen.
Okay, fantastic. Thank you so much for this invitation and for considering me to give this talk in the 6th National Workshop on NEMS and MEMS Theranostic devices at IIT Guwahati. It's an extreme pleasure. I want to thank Arun, who was, of course, with me, a postdoc, and doing great there, and look forward to seeing him and, of course, all of you in person at some point. So as you mentioned, I'm going to talk about halide perovskites, but over this last day, I was thinking about what to really talk about, and just given that this audience is fairly broad, I decided to kind of tailor this talk more towards talking about opportunities and challenges that lie ahead for this new class of materials, which are known as halide perovskites, for light to energy conversion technologies. And of course, when we talk about light to energy conversion, we are typically referring to things like photovoltaics, looking at things like conversion from solar energy to fuels, to chemical bonds, and things like this. And so I will talk about that, and I will give you sort of a broad perspective of, the techno-economic analysis of this, the broader problems that lie ahead, what are the successes that this technology has had, and finally talk about also what we've done in our journey through this interesting material. Before I start, I wanted to thank the funding which has supported this work: Department of Energy, Army Research Labs, NSF, NAVAIR, and of course Rice University Startup Funds. We just recently moved there about 2 years back from Los Alamos, and so that's where we are located. And so if people who don't know where Rice University is, we are in the state of Texas, and we're really on the coast, and so it is really like the weather and everything is like Bombay. And so if you want to do high temperature and high humidity measurements, this is sort of the right place. It is, which makes it quite challenging for doing photovoltaic devices, as you will see that these perovskites are quite unstable in humidity, but nevertheless it's a great place. It's almost almost the weather is like India, and it's a very multicultural city. Houston is the 4th largest city in the United States, and so it's quite fun living there. Okay, so the topics for today, I'll start with talking a little bit about techno-economics of photovoltaics, because I know I see that in India and of course across the world, there's a lot of installations of photovoltaic devices and panels. And I think that looking at just simple techno-economic analysis of what has been done in Europe, we have to be a bit careful. Then I will talk about the physics of photovoltaics. Just a couple of slides. I know that people are from a very broad audience, but if you're doing bio research, then maybe you can take a nap, or maybe you can feel proud of the friend who's doing device physics and semiconductors. But I'll talk about that a little bit, and then tell you why are halide perovskites so special, and what has really happened in the last 5 to 10 years that has triggered so much of interest in this material. Then I will talk about what are the challenges with perovskites, and specifically one of the biggest challenges lies in its stability. When you are talking about photovoltaic devices, you want something that is going to last for 20 years on your roof, and so this is one of the biggest issues that exists right now for halide perovskites. And so I will talk about our efforts to overcome this. And then along those lines, I will talk about a new subclass of materials, also a perovskite, known as two-dimensional perovskites, which bear a lot of similarities to a lot of the two-dimensional materials that we have. But we think that this is really a pathway for achieving high efficiency and durability in these systems, especially for commercial viability. Okay, so let's start. Let's start with this very broad picture. This is really from the DOE Nano Summit in 2009, and this was Rick Smalley, who was a Nobel laureate at Rice, his vision. And these are sort of the 10 things that he pointed out were what he thought were problems in the next 50 years. And I think he couldn't have been more right. And you can obviously appreciate energy, but you can also see this number 8, there's diseases, right? And we are really in a pandemic, and that's the reason I'm giving this talk, in this manner, really sitting, thousands of miles away. And so obviously it is critical that, when we are thinking about powering a planet, this has to be done using sustainable and clean energy as we go forward. And I think if we are able to do that, then we'll be able to resolve a lot of these issues that are there which are listed here. And so let's begin with maybe a slightly not the most positive analysis. This is a case study that I did, and this is data from 2012, and this is pertaining to Germany, and this is Germany electricity generation. And you can see this is the pie chart, and there's different sources of energy that are deployed. And you will see that Germany spent about 100 billion euros to install about 40 gigawatt per year capacity of solar, right? Which is kind of what a lot of governments across, and of course in India there's a lot of this going on as well. And so it turns out that I don't have this chart, but that 40% of this electricity from solar. They get about 40% of electricity from solar, but this only occurs at 12 o'clock at noon, right? 12 noon in the month of May, right? Germany is in the northern hemisphere. It's quite cold there. The sun doesn't shine, all throughout the year as consistently. And so it turns out that the average per year electricity production is just about 5 to 7%. And so if you consider, the geographic locations across the world, different climates, a lot of this kind of an analysis could be done for a large part of the world, right? And so when you're trying to think about subsidizing solar photovoltaic devices in over 25 years, one has to be slightly cautious and really come up with a good business model for deploying solar. And so that's the challenge that lies ahead in India. And I believe that in India, we shouldn't have that much of an issue because we really are blessed with a lot of sunlight across. And so if this is done correctly, then this would resolve a large part of the energy problem in India, and through, of course, sustainable means. Okay, so I think that there is everyone has a list of what are going to be the future technologies that are going to be relevant, and I have my list. And so if you are a student who's working or a postdoc working on solar energy, then I think that in order to be cost competitive and in order to be technologically relevant, you would have to make devices which are greater than 30% efficient. And these may be single junctions or tandems or thermal photovoltaics, whatever this is, but it has to be at least 30% efficient. And this is because if you look at this equation, which is a simple equation of cost of electricity, and you'll see in that equation here, there's materials costs and system costs and things like this, but the efficiency sits in the denominator. And so if you have a big number there, then you will drive down these costs quite significantly. Another technology or another area of research that I think is going to be extremely relevant is solar to fuels, or using sunlight to break bonds or to create bonds in order to for energy storage. And so one example is, of course, that you can split water using sunlight. And if you can do that with just sunlight as the only input, then you will be able to generate hydrogen and oxygen. And hydrogen is of course, there's a lot to be said about the hydrogen economy and things like this. And there's a lot of work being done right now in the United States and of course in Europe on building or trying to develop hydrogen as a source. And the nice thing about these types of technologies, or even for CO2 reduction, CO2 to fuels, is that you overcome the issue of seasonal storage, right? If you think about batteries don't do very well when the weather is extreme. When it's too hot, which is how it is in India, or if it's too cold, battery performance starts to degrade, and so that's not a great solution. But I think if you could do this and store energy in bonds, then this would be a game changer. In addition to this, there's other things like powering the Internet of Things that will require portable power, and so you will need photovoltaic devices, panels that are flexible, perhaps, and of course for water is going to be a huge issue. So solar energy to drive pumps, which is already being done, but things like this are going to be very relevant, I think. So that's my list. And I think as a student or as a researcher in this area, if you're thinking about what are, it's really important that you think about what are the key directions that you want to pursue. And at least my take on this is that you want to do something which is technologically relevant, because you are at the end of the day investing a huge amount of time and effort and money into doing a PhD. And so so I think that these are at least some directions, at least in my mind, are going to be quite relevant going forward. Okay, so let's talk about some physics, and I promise you there won't be too much, just a little bit of physics. And because we are talking about halide perovskites, and this is almost synonymous to talking about solar cells, because that's what they are really famous for. And so I want to describe what a photovoltaic device is, right? And a simple definition, at least my definition of this, is that it's any material, a semiconductor that absorbs lights and uses selective contacts, right? And so what do I mean by that? Which means that if I have a material which can absorb sunlight, right, it has the right absorption or the right band gap, And if these electrons and holes that you're creating, these charges that you're creating, you can separate them such that a positive charge goes to in one direction to one type of contact, and a negative charge goes to another type of contact, then you have photovoltaic action. And so what this would look like is shown here in the current density versus voltage curve here. And it's basically a diode, right? You can see that this is like a diode curve, at least people who are doing semiconductors or electrical engineering. And when you put light on it, you start getting photovoltaic action, and you get two things. You get a short circuit current, which is denoted by this JSC, and then you get a VOC, which is the open circuit voltage, right? And if you take the product of these two, then you get a power, which is shown here as the MPP, which is the maximum power point. Right, and that's what your power is. And so there are basically three aspects to a solar cell. There is a short circuit current, which is quite easy to understand. You have a certain material, it will absorb a certain part of the solar spectrum depending on what the band gap of that material is. You will create electrons and holes, and if you can collect them before these are lost, through recombination, then you'll get a short circuit current. The other two, right, the open circuit voltage and the fill factor, they are quite involved, and these are completely thermodynamically determined. Okay, and so let me share a couple of slides with you and tell you. In fact, also the third thing, which is the fill factor, which is defined as this ratio of this inner blue rectangle, right, to the outer rectangle. And what is it saying is that, you have a maximum theoretical power conversion efficiency, which means the maximum voltage and the maximum current. But obviously you don't get this. What you get is something which is, you lose some energy, and I'll talk about this very briefly. And so the ratio of this inner rectangle, right, area of the inner rectangle to the ratio of to the outer rectangle, which is this green line here, that gives you the fill factor. Okay. And so let me spend one more slide on telling you a little bit more about, the physics of voltages, because, this is very elegant physics or chemistry or whatever you may think about. It's really thermodynamics. And this is where it comes across. And I want to just spend one more slide on this. So obviously everyone has heard of the open circuit voltage, and this may be in different sort of forms. VOC is the same as if you're an electrical engineer or physicist, you'll talk about this as quasi-Fermi level separation, or IMREFs is another definition. Or if you are a chemical engineer or a chemist, you'll talk about chemical potential or free energy, but really it's the free energy that's available to you. And so you can write the second law of thermodynamics in a way such that you have, your photon free energy. That's the energy from the sun, right? You get certain amount of energy that is available to you. Okay. And that's given by H nu minus T delta S, right? S is the entropy, T is the temperature here. And so what I can do is I can write this equation in a much simpler way. I can write this in this context as photon energy is given by the band gap. Eg is your band gap of the material. So if I have a material, that's a material property. And so depending on what material I choose, that's the maximum energy that's available to me, right? If I choose a band gap of, say, silicon, then that's the maximum, that's the best I can do. And Eg minus K, and I can write the entropy as different possible arrangements, right? This is from statistical mechanics, and this is typically written as a logarithm of W, which is different possible arrangements. And so I can sort of rearrange this equation slightly and write this as, in terms of the operating point voltage, right, that I have, or the energy. And Eg is the total energy that is available to me. That's the best I can do. And then you will see that there are these multiple entropic factors that one has to kind of account for, right? So let's kind of look at what they are. The first one is something which is called entropy due to loss of directivity. Okay, what does that mean? This means that if I have a photovoltaic device, and if I take this device out in the sun, then this device thinks that the sunlight is coming from all across, right? From everywhere in the sky at different angles. But that's not really true. In fact, the way in which this works is that the sun subtends very, very specific it is at a specific angle at every time in, during the day, right, in the morning to the evening. And so it subtends a very specific angle with respect to the solar cell. And so you'll see that there is a loss of photons, right, or entropy of photons, which is associated with that. And so if you actually, if you account for that, right, so if you account for all the different solid angles that the sun subtends with respect to your photovoltaic device, then it turns out that there are 40,000 different angles that it can subtend. And so if you divide this first term, this pi is the radiance that you have, right? And omega s is the solid angles. Then if you take the ratio of this, then you get a factor of about 0.28 electron volts or 0.3 volts, which means that this is something that no matter what material you have to just give this away. This is a loss that you can do nothing about. It's almost like God's gift. You just have to give it back, and you can't do anything about it. So so this is the first loss, right? So it's a big number, right? It's 0.3 volts. So if you have a silicon solar cell, then you already have lost 0.3 electron volts from it. The next one, which can be a big loss, is entropy due to incomplete light trapping. So for example, if you made a solar cell where your light trapping was not very efficient, then you will get losses associated with that, and that can be some loss. The third thing is this free energy loss due to power point optimization. Now, what do I mean by that? Again, if you look at this JV curve, you will see that, according to statistical mechanics, statistical physics, what it says is that you can't have best of both the worlds, right? And so if you want to get a maximum voltage, right, then and if you want to get a maximum current, that's not possible. In fact, you have to compromise, and this is so much like life, right? And so that compromise is what this is talking about. So for example, at voltage, when you are at this VOC point, there is no current flowing through your device. And so in order for a current to flow, you need to give some voltage, right? And that's this red curve here. You can see that there's a loss in the voltage. To reach this maximum power point, you're losing some voltage. And similarly, in order to get a current, you need to give away some current in order to gain a voltage, right? And so that's what that compromise is. And so this can be, in the best case scenario, and this is really the fill factor, right? So you can see now that this is really coming for thermodynamics. It's not a circuit property, as a lot of, there's a lot of misconception in electrical engineering that this is really coming from a circuit, but in fact, it's coming from thermodynamics. And so the best you can do in terms of a fill factor is 89%. And so if a student comes to you and says that he's made a solar cell with 90% fill factor, then he's violating the laws of thermodynamics. Okay, that's not possible to do. Then there is another one which is called free energy loss due to poor quantum efficiency. What this is telling you that, if you are at open circuit, you absorb electrons in holes, and what they should do, because there is no electric field in the device, they should just recombine and give out photons. And if they don't do that, then you're going to lose some voltage associated with that. And then finally, there is some constants, which I won't go into details of. But you can now appreciate and see that there are so many factors that play a very important role in terms of trying to maximize the voltage of a solar cell. And so that's really involved, and quite, the physics is quite beautiful associated with that. And so when you're trying to make a device, this becomes quite important to kind of think about all these factors. Okay, enough of the physics. Now, when we are talking about photovoltaic technologies, then this is something that you have to look at, right? And this is basically a chart of efficiency versus the years. That is maintained by National Renewable Energy Lab, and this covers all different technologies. And so the top two lines, yellow lines or rectangles, correspond to gallium arsenide, which are 3, 5, group 3 and 5 materials, which are the most efficient solar cells that we have today, but they're extremely expensive, cost prohibitive. It's about $50,000 per square meter to produce this material, not very practical for large deployment. What is really feasible is silicon, which is technology of the today. That's the best technology that we have for single junction, and this is about 25% today. Now, there was also this dream that people had since about 30-40 years, is to be able to come up with a technology using dirty chemistry or solution-based chemistry. There was this idea that you could take materials, put them in some solvents, right, spin coat them, and you would get a nice efficient photovoltaic device. And these were referred to as the generation 3 solar cells. So it turned out, as it has it, that over 30 years, this did not quite take off. The best efficiency, the best people could do is about 10%. Okay, and so some examples of these are dye-sensitized solar cell or Gratzel cell. People have been working on this. Organic photovoltaic devices, right? Polymer solar cells. And so, they went along till perovskites came along. And you can see that just at the point where the dye-sensitized PV and the organic PV meet, that's where perovskites take off. And so all the knowledge that was accumulated in processing this material, was now directly translated to the perovskite, which also helped to move very fast in terms of efficiencies. Okay, and so what has happened is this: if you put everything in context, I'm plotting the record efficiency or the efficiencies of different photovoltaic technologies of all the common, well-known semiconductors, okay, that we know of. And you'll see that this is over 50 years, and it has taken a lot of effort and 5 decades to reach these numbers. And in comparison, you will see perovskites, which has had an exponential increase. Just in about 10 years, we are at about 25% efficiency, which is on par with silicon, right? And so obviously people have got very interested. People are very intrigued by what are the properties of this material, and that's why the whole entire world is kind of rushed into exploring this wonderful material. And so now these are recognized as a new class of semiconductors with really this is quite unprecedented just from a historical perspective. And so what are these perovskites, right? Like finally we come to this. In fact, this is something that that's been around for a very long time. This was discovered in 1800s sometime by someone who was not named. Actually, it was not Perovsky. It was somebody else, in fact. But it was named after a person whose name was Perovsky. But his friend discovered this, and he named this mineral after him. And so and so Perovsky got the name, he became popular because now we have perovskites. And so if you ever discover something, please don't name it after your friend. Otherwise, he or she will be really popular and you will have to suffer, through history. So anyways, these are materials. They have a chemical formula of AbO3 or AbX3. And common examples are calcium titanate, strontium titanate. These have existed and people have studied these. And so what these halide or hybrid perovskites are, that what they did was they replaced this A, B, and O cations, right? By A here was replaced by an organic cation, so methyl ammonium. The B metal here was replaced by the lead, and the X was a halide, for example, like iodide. But you can also have an inorganic system like cesium lead bromide or other things like this. Now, in addition to this, you also have a subclass of this, which is these two-dimensional perovskites. And so what happens in this is that you add one more organic cation, right? One more organic chain, which which is quite large, right? So for example, this would be a 4-carbon atom chain. And if you add that, it turns out that it doesn't have any space to go into this octahedra, right? Because it is too large. And so what it forms is a layered structure, right? It forms a van der Waals material, which is shown here on the figure, right? And so you have these same octahedra slabs here, which has 3 slabs here. See, you can see 3 of them, just like what you have on the figure on the left. But now these are separated by these organic interlayers through a van der Waals interaction. And so these turn out to be layered perovskites. Now, in contrast to some of these other inorganic perovskites like calcium titanate, which are hard materials, these are really soft semiconductors. Okay, so the Young's modulus, which is a measure of how hard a material is typically, is 10 times smaller than that of other semiconductors like silicon and gallium arsenide. And the nice thing about this is that you can use simple, solution-based chemistry, dirty chemistry. You can tailor each and every site of this atom, which gives you a lot of flexibility in terms of what you want to do, in terms of tuning its properties and structure. Okay, so what makes them special? So let's look at some of the properties, and I've sort of summarized this for you. First thing is that this really, fulfills the dream of using a low temperature processed material for light to energy conversion. And so, for example, you can see here this beautiful single crystals which are made using solutions chemistry. These are, really large crystals. And in fact, this is where things started off at our end in my group, and of course Arun was also along with a lab that time, doing some really nice work, where we developed this method which was called hot casting. And what we were able to show is that if you do this process of hot casting, where you take a hot substrate and drop these precursors of the perovskites and spin coat them, then you get these films which have very, very large grains. Okay, and so if you have very large grains, these are great for transport. And in fact, if you look at these cross sections, you will see that this looks like bulk semiconductors. And you see this very nice sharp diffraction pattern, right? Sharp peaks. And so this is actually for the first time, at least what I can recall, is that this is the first material where you can use chemistry, solutions chemistry, and get these very nice, beautiful, sharp peaks in the diffraction pattern, which is suggestive that this is an extremely crystalline material. And some of these peaks are such that you actually can actually see in a polycrystalline film, these match very well to what you would see in a single crystal. So you can make this very, very nice single crystalline, near single crystalline like films, with these large grains. And we did this and we fabricated these simple devices. The devices are very simple. You have a perovskite layer, which is the light absorber. And then, as I was saying, that there are these selective contacts. You have a contact which will take positive charges, and you have a contact which will take negative charges, right, selectively. And so we were able to make these very nice, efficient devices. And I won't go into details of this because these are some things which are specific to perovskites. But what we were able to show is that your current density, if you see this middle curve, bottom middle curve, this scales very nicely with the grain size. As your grain size increases, your current density also increases. And in fact, as a result, you get very nice power conversion efficiencies. You get about average efficiencies of 15%. This is in 2015. The best efficiency we got was 18%. But you can see that that's an outlier. That doesn't really sit into this. That's one batch of devices that worked well. And so, statistics are important when you're doing this kind of research. And so that was sort of our entry into the field of perovskites, and we were quite late into it. But this was a great result, which got us a lot of visibility, and sort of things took off. Now, in addition to this, in addition to the fact that these are solution process, there are a lot of other properties which are really interesting. These are direct band gap semiconductors, and so that means that you have, it can absorb, small amount of material can absorb a lot of light. It has a large absorption coefficient, right? Which means that you don't need a lot of material. In fact, the absorption coefficients are greater than that of gallium arsenide. You can tune the band gap of this material using solutions chemistry. You have small effective masses of your electrons and holes. And so what this means is that these electrons and holes can really move quite freely across these bands, right? This is representative of the slope of these dispersion curves, or the parabolic bands that you have. And so you have good transport. You can really create free carriers, which means these electron hole pairs are not bound together. And perhaps the most interesting property of this is that, of course, this material has a lot of defects and grain boundaries, as you could see in the optical images. But this material has a very unique property that it is defect tolerant, which means that these carriers, right? So this is a plot which is showing your photoluminescence as a function of time, right? And it's showing you that these carriers, if you make a film in a certain way, they can live in this material for a very, very long time, which makes it very easy for when you're making a device to extract these carriers, right? And this is very unusual. And or rather, the most unusual things about this material, or the mystery of this material, is that all of these properties, right, exist in a single material. Okay, and these are just amazing properties to have if you're trying to make a device from this. And so that's kind of the most amazing thing about these perovskites, which makes them very special. And so we have a lot of projects in our group, that we are working on, all kinds of things. Going from solar cells to photocatalysis, LEDs, electron sources, radiation detectors, and these are some of the papers that have recently been published through the groups. Okay, Now, all this is great, but now I will talk about, some not great things. So one of the challenges with these halide perovskites is that, these are not very stable materials. And so if you make a photovoltaic device using this default material, which is this methyl ammonium lead triiodide, it turns out that when you put light on this material, you get a degradation in the current, which is shown in the plot on the top right. The voltage remains fine, but the current keeps degrading over time. And then when you turn your device off or you turn your lights off, and then you come back, you again restore your device, but you keep getting this sort of degradation. And so this is obviously not good. If you're trying to make a photovoltaic device, this is not what you want. And so this has been a challenge, and there are a lot of reasons for this. Things like ion migration, light induced, polaron formation. I won't go into details of what the physics of this is. But obviously the world is kind of realized this, researchers have realized this, and there's been a search for getting a three-dimensional perovskite which is technologically relevant, right? Technologically stable. It exhibits stability which is technologically relevant. And so let me tell you what we did along these lines. And so one of the things that we did is that we said that, this methyl ammonium, instead of having a single A side cation, let's replace this with something which is more complicated, which looks like this, which you have in formamidinium. Fa is your formamidinium, Ma is your methyl ammonium, and cesium, small amount of cesium. And if you do this, then it turns out that you're able to get a nice cubic phase of your perovskites. Okay, and this is quite stable. And in fact, it also exhibits very good temperature stability. So this is, looking at X-ray diffraction. This is just a two-dimensional plot done at a synchrotron source, and I'll talk about that just in a little bit. But if you look at an X-ray XRD pattern, you will see that these peaks, the 100, 200 plane, they're very stable with temperature. They don't change at all, just showing that this is a nice and stable material. It has a nice band gap of 1.5 electron volts, which is shown here on the red, and nice photoluminescence, very strong. And so it's very similar to the material you made, but it's just that it is much more stable. And so what we wanted to understand is that what happens when you put light on this material, right? Light and electric field, which is what you want, because you want your photovoltaic device to last for more than 20 years. Then you want to understand what happens with continuous light on this material. And so people have been doing a lot of work on this. Again, I won't go into details of this, but what was not done when we started doing this is that there were very few measurements which were able to correlate what happens to the structural properties under device operation, right? When you make a device, when you put light on it, what happens to the structure? Does it degrade? Where is the degradation coming from? And so we wanted to understand this, right? And so we performed these measurements as a synchrotron beam line at Argonne National Lab. And the experiment was so forth. We took a device, or we took materials. Basically, we took three types of samples. We took single crystals, we took thin films, we took thin film devices. And we mounted a solar simulator on the top. And at the same time, this device was in operation, and we were measuring the X-ray signature, which is shown here on the right, this panel, this blue panel here. You see these, this pattern here, which corresponds to the X-ray diffraction, the lattice spacing, right? And you can take a line cut of this and monitor this as a function of light. And so here's what this curve looks like. If you take a line cut vertically across this, you see these diffraction line, this diffraction pattern, right? These peaks which correspond to the D-spacing, the lattice spacing in the material. And what we found, or what we discovered, was a very interesting and intriguing effect. We found that light, we saw that when you put solar light on this material, you get a lattice expansion along all the planes. Okay, so which is shown here on the bottom plot here. So this is showing you the 100 plane. You see that it is going towards a lower Q value, right? This is the wave vector, which is inverse of the lattice of the real lattice spacing. And so if you take the inverse of this and actually plot the lattice parameter as a function of time, you will see that your lattice parameter, so lattice is actually parameter is increasing, it's expanding as you put light on this. So it was very interesting. And so we wanted to understand what happens and what's really going on. And so we did more analysis. One of the things that we found is that this is a reversible effect. When you put light on it, you can, and you take the light away, it can come back to its original position, which is shown here in the dotted line. But another interesting thing was that if you look at the full width half maximum, right, the width of this peak, it seemed that it was that as you light soak this device, that this full width half maximum decreases, right? Which means that you're making these peaks, these peaks are getting sharper, which is indicative that you are increasing the crystallinity in this material. So light leads to expansion of the crystal, which leads to increase in the crystallinity of your material in this case, which was quite interesting. And so obviously we wanted to understand what happens to a photovoltaic device. And so we then measured the photovoltaic response simultaneously. And this is what we find. So here's, for example, the changes in the open circuit voltage, the fill factor, and the short circuit current. And you can see, I'm sorry. You can see that your voltage and your fill factor increase quite nicely. This current also increases somewhat, but you see this increase in this. And in fact, if you plot the change in the voltage as a function and the change in the fill factor as a function of change in your lattice parameter, right, this delta D over D0, you see that both of these effects are synchronized, which means that you put light on the sample, the lattice expands, it increases this crystalline quality, which leads to an increase in your voltage and your fill factor. Okay, should I continue? Okay. Okay, so and of course this effect is plotted here, right, which is shown here in your dark and light curves. This is your JV curve, which is in blue. And after you put light on this material, right, you see that there's an increase in the voltage, which is sort of shown here. And so by doing this, we were able to get a device which goes from 18 to 20%, which is a big change, right, big increase in your performance of your device. So this is all great. And also this material showed pretty promising stability. You will see that this current now is quite stable. You see it's pretty flat over about 1500 hours under one sun illumination. Also quite stable under 10 suns and also under some other extreme conditions. So we were able to sort of show that we can increase the stability by doing, making this very complicated triple cation 3D perovskite solar cell. But of course, we were not very happy with this, and we wanted to sort of come up with a different solution. And one of the other things that we wanted to try to do was think of alternative strategies. And so one of the things was to look at this subclass of these perovskites, which is these two-dimensional perovskites. By the way, how much time do I have? If you tell me, then I can sort of I can stop and but let me know if there's how much time I have.
Sir, am I audible?
Yes, you are.
Yeah, so the session was supposed to run till 11:45. So you still have 4 minutes around left. That is including the interacting session.
Okay, all right. So maybe I should, I'll just talk for another minute or so, and then I will stop, and then I'll be happy to take questions.
Okay, sir, sure.
Okay, thank you. Okay, and so, one of the things that we were now beginning to look at is these two-dimensional perovskites that I showed you before. And what we were able to show is something quite interesting, that if you measure the stability of these two-dimensional perovskites, which is shown here, and you compare the stability to the three-dimensional perovskites, right, you see that these 2D perovskites are dramatically much more stable than the 3D perovskites. Okay, both in terms of continuous light and also in terms of humidity, right, 65% humidity. And so what we were able to show is we were able to get about 12% efficiency. So they are not as efficient as the 3D, but the stability of this material is quite much superior, and this is the intrinsic stability of the material. And so this was sort of a big result that we had, And so now we are going through efforts of trying to come up with a 2D perovskite which is more stable. And I'll skip this part, but I will give you the final result. And so where we are at really is that now we have a 2D perovskite which has about 17% efficiency, and you can see that the stability is very, very good. At about 70% relative humidity, we have about T80, which corresponds to the 80% of the lifetime. We have about 7,000 hours. Even under 5 sun illumination, this is very, very stable. And in fact, if you compare and if you look at sort of where we are going with this, this is really the trajectory. This is the work that we've done, but this is the path forward. And I think that if we are able to hit this number of 25% and demonstrate this, retain the stability, then there would be a real clear path for commercial viability. Okay, and so I will stop here for now, but I want to just thank my team, which has done this work, and again, funding and also collaborations, collaborators, Professor Jackie Even, Mercury Canadzites, and Claudine Catan, who've really contributed heavily to the efforts that I've talked about today. Okay, and I'll be happy to take questions.
Thank you very much, Professor Mohite. That was an interesting talk. So now I'll be reading out the questions from the participants.
Sure.
So our first question is, how grain size increases the power conversion efficiency?
Yeah, so typically when you think about a device, right? So let me see if I can show you this one more time very quickly. Yes. If you think about this device here, right, if you can see my screen, right, if you have a grain, right, which is so this is a representative image, right, this thickness actually, right, of the perovskite is about 500 nanometers, right? You need about 500 nanometers of the perovskite to absorb all the light. Now, if I have a grain size which is very small, right, grains are running across this, then if I have an electron and hole or charges which are going across this, the chances are that they will run into grain boundaries, right? And you don't want that. So if you have a large grain size, imagine that you had a single grain across this entire device, right? Then you would have no recombination. You would not lose these carriers to the grain boundaries, and this would lead to an efficient sort of current and voltage that you have, right? So that's how grain sizes work. And so what you want is a very large grain size, as large as possible, so that you avoid recombination to the grain boundaries. And typically any of all these defects and grain boundaries are generally not good for transport, electron transport.
Thank you, sir. Our next question is, why lattice expands when we put light on the material?
Yeah, that's a great question. We don't have an exact mechanism of why this works, but the working hypothesis is this, that, if you think about the band structure of this material, or if you think about bonding and anti-bonding, which you may have learned in chemistry, you have an anti-bonding orbital, which is iodide, which forms the valence band, and you have a bonding, which is the lead, which is the conduction band. And so when you get a transition there, right, you have an electron which goes to the conduction band. What you've done is, because you sort of weakened the bond, the lead and iodide bond. And so as a result, what happens is that your overlap sort of decreases and you start to get some sort of an expansion, right? So that's sort of the working hypothesis that we have. And so now, additionally, when you have a charge on the lattice, right, when you put light on this material, you have electrons or holes which are present, which lead to some sort of a local strain, which leads to an enhanced effect of this expansion. So this is kind of like a working mechanism that we have, but of course we don't know the exact mechanism behind why this happens.
Thank you, sir. One of our participants, Mr. Atanu Purkasta, is raising their hands. I would like to request him to unmute himself and pose the question.
Sir, am I audible?
Yes, you are.
Okay, sir. My name is Atanu Purkasta. I'm a research scholar from Department of Electronics and Electrical. I'm working under Dr. Arun Tej.
Yes.
on lead-free perovskite solar cell. So I have two questions, sir. Actually, sir, it is what I have seen, sir. It is very difficult to get a good film using anti-solvent technique in tin perovskite solar cell. So can you tell me, sir, whether hot casting technique will be good for 2D and 3D tin perovskite? One question, first question. And second, sir, what are the steps we should follow to get a good efficiency and stability for tin perovskite solar cell that we can compare with the lead perovskite, sir?
Yeah, those are two good questions. I think so. The tin, of course, is a very challenging, right? Lead free is a challenging area of research, right? Because as you know, this tin tends to oxidize very fast, right? And in fact, there is a lot of challenges in terms of how you process tin. And in fact, there are some very nice papers that have come out more recently, and I can forward, maybe in a separate, if you send me an email, I can forward them to you. And I think they use different combinations of course, the anti-solvent approach, but also using some molecules to passivate surfaces and block the oxidation from 2 plus to the 4 plus state for tin. So I would say that you should follow that. But in general, and of course, I'm guessing that you're doing this in a glove box, but that's the environment of the box is extremely relevant, right? So that's important. And and I think you could try hot casting. We have not tried it, but I think you could try that out and see how that works. I think in general, what we have seen is that helps, right? Whether that's a scalable technique, I don't know. And I think your second, did I answer your questions, or did you have one more? I think, right?
No, sir. No, sir. That's all, sir. That you gave me the answer, sir. Okay. Thank you, sir.
Okay. Yeah.
Thank you, sir. Our next question is from Mr. Ritesh Kant Gupta.
Yeah.
So he asks, looking at the recent reports, 2D perovskite solar cells have reached over 18% and still maintaining their stability. So is it possible that 2D perovskite itself can have efficiency greater than the 3D perovskite solar cells with more stability?
Yes, in fact, that's exactly the direction we are going in. In fact, the state of the art 2D perovskite efficiency now depends on how you define a 2D perovskite, right? And in fact, that's the part which I was not able to cover. But oftentimes, when people are reporting the 2D perovskites, right, they're not really 2D. They're talking about a mixture of 2D and 3D. And you can kind of see that they're not pure. Okay, and so you have challenges. As if you have more impurities, you'll have challenges just like the 3D. And so in fact, we think that the way where we are coming from is that we are making a case for pure 2D perovskites, where we really think that if we are able to hit an efficiency of over 20% while maintaining the kind of stability that I showed you, right, which is really the state of the art today, right, in terms of perovskites, then we would have a great chance for building a technology based on that. So, but of course, you have to be careful. Just to answer your question in the beginning, that the 18%, you have to be careful whether that comes from a 2D perovskite, pure 2D, or does it come from a 3D 2D. And the way you can tell this is by looking at the open circuit voltage. Typically, in a 2D perovskite, you get a band gap of about 1.7 electron volts, but if you look at the external quantum efficiency spectrum, right, that's where it kind of takes off 1.7 eV. Whereas in some of these reports, you'll see that the band gap is very similar to what you get in the 3D. So you have to be kind of cautious of how you're defining this.
Thank you, sir. Thank you very much for the interesting talk and even interesting question answer session.
Thank you.
So we will now be we will now be moving on to our next session. Next, Mr. Shauradeep Dey and his colleagues will be presenting. In the next session, Mr. Sauradeep Dey and his colleagues, Mr. Ashutosh Bandyopadhyay and Mr. Vibhas Kumar Bhunia from Biomaterial and Tissue Engineering Laboratory, IIT Guwahati, under the supervision of Professor Vimandi Mandal, will brief about the basic process involved in the printing of 3D objects using a thermoplastic extrusion-based printer, Ultimaker 3. I would like to request the group to proceed with their presentation.
All present here. I, Sauradeep Dey, and my colleagues Ashutosh Bandyopadhyay and Vibhas Kumar Bhunia from Biomaterial and Tissue Engineering Laboratory, under the supervision of Professor Biman B. Mandal, would demonstrate on 3D printing using Ultimaker 3 at NWNTD 2020. In this session, we would be briefing about the basic process involved in the printing and fabrication of 3D objects, right from the development of 3D models, selection of appropriate materials, slicing of the 3D model, and the printing of the model using a thermoplastic extrusion-based printer, that is Ultimaker 3. We would also answer your queries at the end of the presentation, so stay tuned.
First of all, I would like to introduce you to the basics of 3D printing. 3D printing, also known as additive manufacturing, is the process of layer by layer fabrication of 2D layers on top of each other to form a 3D object. It is basically a manufacturing process that was developed to bring prototypes and 3D models to the physical world using polymers. Moving on to the history of 3D printing. 3D printers were conceptualized in between 1950 to 1970, but a commercial patent was obtained by Charles Hull in the year 1984 under the name of stereolithography, and the commercial printer he developed was named the SLA-1. Later on in the 1990s and onwards, the advancement in technology and lower fabrication cost of the printers, as well as the expiration of patents, popularized these printers and led to many commercial printers which we find in the market now. There are several kinds of 3D printing techniques available in the commercial domain. The first one we would discuss is inkjet printing. Inkjet printing has its roots in the traditional office printing method. This technique has evolved from a slightly modified desk-jet printer that could hold material in its cartridge and fabricate multi-layered 3D constructs. The technique simply involves ejecting the polymer ink from its printer nozzle onto a printing surface while moving in the X, Y, and Z axis. It is a non-contact printing technique that can build 3D structures by depositing droplets in a layer-by-layer fashion. The two most common approaches that are applied in the dispensing process in this technology are piezoelectric and thermal-based systems. The piezoelectric mechanism involves the rapid actuation of a piezoelectric actuator that generates enough pressure momentarily to expel the material from the cartridge onto the print bed. While the thermal approach involves the use of a heating element to raise the temperature of the ink material at the nozzle to reach more than 300 degrees Celsius for a few microseconds, which results in the formation of vapor bubbles that expand until the material is expelled from the nozzle. Here we can see some examples of inkjet printing being used for machine, mechanical parts, and bioprinting. The materials used for this printing technique are mostly low viscosity materials, or they are in their molten state. Moving on to the second technique of extrusion-based printing, it is one of the mostly used and common and promising technique for fabrication of prototypes at low cost. It principally involves continuous layer-by-layer programmed deposition of material through a dispensing nozzle or printing head using either a mechanical screw-driven or piston-driven or pneumatic dispensing system onto a substrate in discrete volumes. There have been modifications in the print heads of these printers to enable electrospinning and microfluidic-based dispensing techniques, which of course improves the resolution, but results in a lower material deposition volume. The fabrication of 3D constructs using this method mainly relies on stepper motors and pneumatic actuators for the movement along X, Y, and Z axis. This method is most commonly used by the common folks as these printers are highly cost-effective, robust, and self-replicating as well. Here we can get an overview of the printing process as well as a few examples which show the utility of this printing technique in various prototyping applications. Extrusion-based printing can facilitate a fabrication of structures using a wide variety of materials, ranging from the thermoplastic polymers to hydrogels and biological materials as well. The next one we move to laser-assisted 3D printing methods, also known as stereolithographic printing methods. Laser-assisted methods or stereolithographic methods involve the fabrication of 3D constructs using a laser for the deposition of polymer ink on a substrate, preferably through a contactless procedure. Laser-assisted printing involves direct laser writing or stereolithographic technique. Both techniques demonstrate high spatial resolution, mostly determined by the area of the laser spot, and are based on the reservoirs for the storage of polymer inks before deposition. Laser direct writing modalities help in the forward movement of ink from its origin, that is a coating on a reel or matrix, to their destination, which might be the printing bed or a substrate, using a powered laser light and an instantaneous heating. This deposition is followed by a cross-linking step. This was an old printing method, and it has been modified in the recent times to develop stereolithographic method. So the stereolithographic method, which is the most commonly used one, for it, it is a format that involves a laser-assisted layer-by-layer cross-linking of liquid polymer in a one-step fashion, which, while moving in Z direction, makes a 3D object. It possesses the advantage of the use of a low-energy laser as well as a safer wavelength of light, while maintaining high printing resolution and faster printing speeds. This process is only limited by the size of the print bed or platform for the dimensions of the print and the requirement of higher polymer volumes to fill the reservoir. So here we can see the working principle of the stereolithographic printer on the left hand side. Here the geometry of each layer is projected layer by layer onto a polymer bath by a laser or LED light source. This cross-links the polymer and leads to the formation of a 3D object on the printing bed. We can see its two different implementations on the right hand side. The first one on the top is the laser-based polymerization platform, which cross-links each layer one by one using a laser. And the second one on the right at the bottom is an LED array-based polymerization system, which cross-links the polymer of one layer all at one time by an LED array and an image of the layer it wants to polymerize and print. These printing methods require a photopolymerizable polymer ink, of course, because the polymerization process would be based on a light source, which is a particular wavelength based light source, such as a UV or a visible light source. So the UV resin or photocurable resin that being used is the raw material that is mostly important for this printing method. Moving on to the next technique of selective laser sintering. It is a prominent technology used in the formation of 3D ceramic, polymer and metal based structures by the use of sintering process to selectively fuse layers on top of each other by a high powered infrared laser scanning the surface of the material to be sintered. The mechanical properties of the sintered constructs can be dictated by the scanning speed of the laser, while the energy of the laser can be harvested and modulated to influence the nature of sintering from solid state to liquid phase. This technology has been mainly employed for the fabrication of prototyping using polymers such as PCl and cellulose, ceramics such as hydroxyapatite and bioglass, and metal alloys such as titanium, aluminum, vanadium, and cobalt chromium alloys, as well as some composites. The prototypes fabricated using this method have higher mechanical properties, durability, and slower degradation rates. These prototypes can directly be used in industrial applications as well. So this is used for the fabrication of industrial parts and chassis, as well as medical equipments and implants. We can visualize the working principle of this printing method on the top right hand side, where a machine part, a gear, is conceptualized and fabricated using a metal alloy by the virtue of laser sintering method. Below you can see some of the printed 3D structures on the right hand side that are printed using this SLS technique, and it also supports a variety of powder materials starting from different metal powders for the automotive industry to polyamides and thermoplastic polyurethane that can be used for biological applications and biomedical applications. All material powders used in this technique can be tuned and altered for specific applications so that we can regulate their mechanical properties as well as their durability.
Moving on to the several steps in the 3D printing process. Here we will be briefing on the extrusion-based printing technique. The first thing before designing obviously is ideation. Based on the idea, one has to formulate the design. For designing, one can either use a 3D scanning technique for 3D scanning a particular object or develop one using various softwares available for designing like Tinkercad, AutoCAD, and several other open source softwares. The file developed using this 3D modeling software is then stored in the form of STL accepted by the slicing software. So this .stl format is commonly known as stereolithographic format. Next, we have to slice the developed CAD model. As we know, 3D printer prints objects in a layer by layer fashion. Out of many available, the one that we will be demonstrating in the later slides is Cura. It is a free software. In the software, one will find a number of options or settings available to change. Each one of them holds some special function. Once sliced, the .stl file gets converted into G-code. As printer only reads G-code, which are instructions for the mechanical movement of the printer and the print bed in a XYZ axis. After the G-code is developed, then one has to choose the fabrication material. Based on the application or model type, one has to choose the polymeric material. This is dictated by the type of application, the strength of the material, or the strength and durability of the particular object that needs to be developed. The polymers that are available in the form of pellets or gels or filaments are mostly used in this extrusion-based printing technique. The polymer filaments which we will be using is thermoplastic in nature, which melts at a higher temperature than the normal polymeric gels that are available and thereafter gets deposited in a layer by layer fashion. Next, we have to set up the printer. Finally, the polymeric filament, after if it is being inserted into the printer, the G-code is loaded into the printer memory. Next, we have to check the extrusion of the filament and initialize the printing process using the loaded G-code. Once the printing process is completed, we take out the model from the print bed using a spatula. After the printing process is completed and the 3D printing structure is being taken out, post-processing sometimes is needed. Post-processing can be done to remove the excess non-required materials as well as the support structures. Alternatively, one can also use sacrificial materials in such extrusion-based printing techniques, such as some support structure for the 3D structures which we have developed, which can be simply removed by dipping into a solvent. Coming to the applications of this extrusion-based 3D printing, so this extrusion-based 3D printing has been used in medical, food, architecture, and automotive industry for the purpose of product development and prototyping. Recently, it is gaining popularity in several other industries such as construction of houses, footwear, and fashion accessories. Similarly, in this slide, we will be discussing about a typical process for extrusion-based bioprinting of 3D tissues. Briefly, the first two steps, that is step 1 and step 2, that is imaging and design approach. Imaging of the damaged tissues and its environment is used to guide the design of bioprinting tissues. MRI or CT scan based approaches and biomimicry are used for the development of CAD models of biological tissues and organs. Reconstruction from a scan could be used for making the design patient specific. Now coming to step 3 and step 4, these are the essential steps in case of bioprinting, that is material selection and cell selection. The choice of biologically compatible materials and cell sources is essential and specific to the tissue form and functions. So commonly, the materials that are used for bioprinting include synthetic polymers such as polyurethane, polycaprolactone, PLA, or PLGA, or some natural polymers such as collagen, alginate, gelatin, as well as decellularized ECM. The cells could be sourced from patient or from a compatible donor according to the application. Finally, the step 5 is bioprinting, that is printing the cells and the materials all together in order to create the specific CAD model that we had developed. So all of these components need to integrate within the bioprinting system. Sometimes the application of these are different. Sometimes the bioprinted 3D tissues need to undergo maturation before their end use, or the 3D printed tissues may be used for in vitro application or in vivo implantation. We will now discuss the steps for 3D printing in detail, followed by a demonstration of the same using a filament-based thermoplastic extrusion printing system, Ultimaker 3. There are several imaging techniques. For example, CT scan is used to
We will continue the session in 5 minutes.
demonstration of the same using a filament-based thermoplastic extrusion printing system. There are several imaging techniques. For example, CT scan is used to capture data of an inside organ and at different level of density of the human body. It captures detailed information of inner parts of the body with the help of X-ray beams. On the other hand, MRI is used to capture detailed structure with the help of powerful magnetic field and radio frequency pulses. So the CT scan or MRI images are stored in the form of DICOM images. DICOM, that is Digital Imaging and Communication in Medicine, is the international standard for medical images and related information. It defines the formats for medical images that can be exchanged with the data and quality necessary for clinical use. Now coming to step 2, these DICOM images can be then processed into open source softwares like 3D Slicer using image segmentation to reconstruct the 2D images into the 3D model. After the 3D digital model data is produced from a 3D scanner or other medical imaging techniques like MRI and CT scan, it can be customized or tweaked according to the end user usage. Further, the obtained 3D CAD models could be refined before slicing or designed from ground up using softwares such as CATIA, Maya, Autodesk 3ds, SolidWorks, Tinkercad, TurboCAD. So these are some of the softwares that are available over the internet. On the left hand side, you can get an overview of the two most commonly used softwares, that is AutoCAD and Microsoft 3D Builder. 3D Builder is a facile software that could be used for minor modifications and simple 3D modeling, while AutoCAD could be used for detailed modeling and alterations. All the softwares that are mentioned over here have their own advantages and disadvantages. Some of them have more flexibility in terms of functional and modification tools, giving it a complex user interface, while others have a comparatively simple and user-friendly user interface, especially for beginners.
So here we show you how to convert a 2D image into a 3D object using 3D Builder. So 3D Builder is a very utilitarian software that we get with Microsoft Windows 10. So first we will insert the 2D image that we want to convert into a 3D object by the load image function. So here we choose the IITG logo as an example and we open it. Next we choose the options that we want so that the 3D object that we are going to make from the 2D image fits according to our requirements. So we play with different options that we are having in this and we import the image as a 3D object. Once we have imported the object, we reduce the size of it according to our needs. Here we can see that the initial size is 82 mm by 82 mm, which is a huge model and will take a lot of time to print. So we take a scaled down version of it and take it to 25%. And here the different dimensions can be scaled down individually on each axis. And we see that we are able to reduce the size of the object according to our need. Then we will be settling down our object on the floor and we will insert a build plate adhesion surface for the object. Since this is going to fit on the build plate and it is necessary for the object to fit over it, we will reduce the size of the cube that we have inserted according to the size of the object. So the object is 20 mm by 20 mm structure. So we reduce the size of the build plate adhesion cube to 20 mm, so somewhat greater than 20 mm. So 22 mm is unnecessary size that we are going to use. And the height, we can be setting it to a lower dimension such as 2 mm, so that we don't waste the material that we are going to use. Then we settle it on the build fluid and next we change the coordinates of the object to 0, 0 so that it comes near the build plate adhesion structure that we have already built. Now we snap the object over the build plate adhesion structure like this and it sits over it perfectly. Next, We will be saving this subject into the format of an STL file, which will be read by the 3D printer slicing software Cura. So the STL file is saved, and here on we can proceed towards the slicing process. So the next step, after we design the IITG logo, we will have to slice it for the printer to print. So we will load the 3D object that we have currently designed, and we will print it after slicing. So the slicing can be done using Ultimaker Cura, because we are using an Ultimaker 3D printer and it fits all the parameters according to the printer. So we will load the object into Ultimaker Cura, and thereafter we will decide what parameters we are going to use for 3D printing. So we will set the model to have different kinds of infill densities, infill patterns, and the different kinds of layer heights according to the resolution as well as the refinement of the model that we need. So the layer height dictates what kind of smoothness we will get in our model. Here we have set it to 0.15 millimeter, which is 150 microns, which will reduce the layer height to 150 microns, and we can increase it, but the model will become more and more rough as we use larger layer heights. The smaller the layer heights, the smoother the model. Next, we will determine the infill density that we are going to need. Here, we will be setting it to 40%, and the infill pattern, we are going to set it to grid, because there are different kinds of infill patterns available here, and grid is one of the most efficient infill patterns that we could use for making 3D objects that are solid and which can be used for structural components. So here we are going to use a grid 3D infill and 40% infill. We will show you how much the infill changes by changing the infill density later. And thereafter we will see if we need a build plate adhesion. Since we have already used and made a 3D build adhesion platform below the model that we are going to print. So we are not going to use a 3D build plate adhesion, but we have a lot of options such as a skirt, a brim, or a raft. So these are different kinds of 3D build plate adhesions that can be attached with our model and used for the adhesion of the model on the plate while we are printing, because some of the times the model might get dislodged while printing at higher printing speeds. But here we are not going to use any build plate adhesion due to the already built attached build plate adhesion. And thereafter we are going to slice it. As you can see, there are a lot of more options that we can change according to the printer specifications that we have, and we can choose the model material that we are going to use. So here we have chosen PLA, but we can choose different kinds of materials according to our availability, and the kind of print core that we want to use determines the nozzle diameter that we are going to use for 3D printing of our model. So here we have chosen a 0.4 print core, which will extrude the PLA and print our model. So the slicing is already done, and we can see the preview of the sliced model. Let me zoom it and show you how it looks from the top. So we can scroll through the different layers, and here you will be able to see the infill that we are going to do. So the grid infill looks like this, and at 40% density, it looks like this. At higher densities such as 60% or 70%, let's set it to 60% and slice it again, and we can see the amount of infill that has increased. It increases drastically, and the infill density determines the strength of the model that you are going to get. So if you want a stronger model, a more solid model, you can use a higher infill density. There are different kinds of infill patterns as well, and you can use different infill patterns for getting different kinds of strengths. But grid is shown to have the highest strength among all the kinds of infill densities, other than the honeycomb structure, which is also a different kind of grid structure, which gives high strength structures at low infill densities. So once we are done with the infill density, we can slice the model. So let's set it back to 40% and slice the model. Now there are two options. We can either save it to a file, a removable device, or if we have a printer connected to the software Ultimaker Cura, we can directly send it to the printer for printing. But since we are not connected to the printer yet, we will be saving it to a removable device for printing it later. So you can see that it has been saved. And next we are going to 3D print the model from here. Similar to the IITG logo, we would slice the model of a human ear. First we Cura and load the 3D human ear STL file that we have. It is a reconstruction of an actual human ear, but we are going to print a smaller version of it for the demonstration purposes. First, we scale down the model that we already have using the scale down function in Ultimaker Cura. So we can set it to whatever percentage we want according to the size of the print that we want. We will set it to 50% for our need and see the dimensions of the model according to the percentage of scaling down that we have done. Thereafter, we would choose the options that we will need for printing this similar to the IITG model, and here we would choose a layer height that will be more fine and the model will be a smoother one, and the infill density that we are going to use is 40% and the grid pattern would stay. Similarly, the build plate addition would not be there as it is a fairly large model which can stay on its own on the build plate and we don't need to provide any build plate addition to it. Thereafter, we can select the extruder that we are going to use. We are going to use the first extruder of 0.4 mm AA cartridge and we are going to slice it. After the slicing happens, we will be able to see the layers as we did previously. So we see the preview of the model and we can see the infill happening at the layers of the middle, and the top and bottom layers are obviously solid infill because the object should stay and look like a 3D object, though the inside is a little bit lesser infill. Thereafter, we can save it to the removable drive. We can see the expected time and the expected amount of material that we would be needing for printing this on the bottom right hand side. We can save it to the removable drive and we are good to go.
Coming to step 3, material selection. In this section, commonly available materials are compared and evaluated based on several performance matrices. Materials must be suited to the application in order to have successful results. The properties of any material become increasingly important as a product progresses from a concept and functional prototyping to production. It is the combination of the material and the process that dictates the characteristics. Generally, one or two material properties distinguish an additive material from all others. The recommended approach for selecting a material for 3D printing is to first define what properties are critical. So here in this list, you can see the various properties that are taken into consideration while choosing a particular polymer for printing. For example, from the list you can see nylon carbon fibers have greater flexibility than polymers like ABS or PLA, while polycarbonate and high impact polystyrene have greater strength than nylon and PLA. So based on these properties and application, one has to select the suitable polymer for printing. So coming to the material selection for 3D bioprinting. In this section, we would discuss about the currently available biocompatible materials. Selection of biomaterial for extrusion printing involves comparison and evaluation based on several performance matrices, including their compatibility with different bioprinting systems, bioprintability, resolution, affordability, and several other cellular compatibility parameters. Extrusion-based bioprinting has the greatest flexibility among existing bioprinting modalities due to the printing mechanism. So as you can see from this list, extrusion-based printing has the greatest flexibility. Extrusion-based printing has facilitated bioprinting of hydrogels, live cell aggregates in spheroid strands and cell pellet form, decellularized tissues, and micro or nanoparticles. The bioprintability of hydrogels is superior to that of other bioink types. As long as the cross-linking mechanism is incorporated into the layer-by-layer fabrication scheme, bioprinting of hydrogel is relatively straightforward, as other bioink types require additional processing and customized systems for the extrusion setup. So coming to the final step of printing. After the G-code has been fed to the printer and the material has been loaded, we start the process of 3D printing. Here on the left hand side, you can see the use of extrusion printing for prototyping and bioprinting, while on the right, we can see the SLA and droplet based fabrication processes. So now we move on to the demonstration of 3D printing of the previously developed CAD model using Ultimaker 3.
So this is the Ultimaker 3. Here we have two extruders, a 0.25 mm extruder and a 0.4 mm extruder. Though there are many different kinds of extruders available with this, but we are going to use these for the demo. Then we can see the heated glass bed that the printer prints on. This can be removed for easy removal of the model. This comes bundled with the printer, so we are using this today. Then we can see the guide rails for X and Y axis of the extruder. The extruder moves over these, and the Z axis screw can be seen, which drives the bed up and down. All of these are driven by the stepper motors that are available here. Then at the back of the printer, as we turn it around, we can see the filament holding area, the filament loading and holding area. The filament loading docks are labeled 1 and 2 for extruders 1 and 2. So there are two filament holders available. This is how we load the filament into a filament loading dock. Now we move to the front end interface. This is the front end interface of Ultimaker 3. Here we can see three different options: print, material/print core, and system. So first we will go to print option. And here if we insert a USB, it reads the files that are stored on the USB, the G codes that are stored there for printing, and we can use it. So here we can see the different files that have been stored. Now if we go to the main menu and go to the material or print core section, there different kinds of materials can be selected and the print cores can be selected, and the materials in the print cores can be chosen or changed and loaded and unloaded. So in material 1, as you can see, there is an option for load, unload, or changing the material. Similarly, there is an option for print core selection, where we can change and load and unload the print cores and set the temperature of the print code. Thereafter, in the third menu of the system, there different kinds of customizations can be made and different kinds of connectivity options can be found. So we can connect the printer through a network. We can change the build plate temperature, the print head temperature, and the frame lights can be changed. So later on we can use the maintenance section and the language section for different options. Now we will be loading a new material into print core 1, that is the PLA that we are going to print with. So this is how we load the filament into the feeder for extruder 1, and once the feeder is loaded, we will be moving to the front panel. Next we click on load, And now movement of the print head can be seen for loading the filament. Here we can see the filament 1 has started heating up for the loading process to go on. The print core is heating for the material 1 to be loaded. So now we can see the filament loading into the extruder, and we can see the filament coming out of the extruder in a moment. Since the PLA filament has started coming out, we will click here on confirm. And the print core goes to its original position. Once we have loaded the model onto the pen drive or USB thumb drive, we can put it in the printer and we can select print. So here we have saved it under the name year to print. We select the print and the printer starts preparing for the printing process. First it will heat the print core and the print bed. Thereafter it will start printing. So we will show you the printing process in a while.
The Ultimaker does active bed labeling before printing every time it prints. So here we can see the Ultimaker perform an active bed labeling here. So as the print progresses, the Ultimaker prints layer by layer the ear design that we have given. Since it will take more than an hour, we will be speeding up the video a little while and show you the end product after some time. Here is a close-up of the printing that is being done. We can see the first few solid infill layers at the bottom of the ear that we are going to print. They have already been completed, and we will proceed to the mid portion. So here we are at the midway point of the printing procedure, and we can see half of the model has been printed. Since we are at the halfway point, we will be able to see the infill that we are doing. Here we can see the grid infill being done inside the model. We are at the end stage of printing, and the print is going to be done soon. Thereafter, we will show you how to take out the model from the print bed. And this is how we take out the print by scraping using a spatula. So similar to the ear model that we printed, we will now be printing the IITG logo that we made a while ago. So the print has started, and as we can see, the first few solid layers are being printed. After this, we can see the solid platform is going to be completed, and the actual logo has started to print. This is the middle stage of the printing, and this is a very short print. So we will be speeding it up and we will be showing you the final result after a while. We will take out the model similarly from the print bed here as well. So the print progresses, and as you can see, the print is about to be finished. So this is the final end result. So these are the 3D printed models which have been taken out of the print bed post-fabrication. As you can see, there are different patterns over the models which are made due to their layer by layer fabrication and the layer height being 0.15 mm. The fabrication method of extrusion-based printing leads to such artifacts, and these artifacts can be removed in two ways. The first one being they can be developed using solvents, and they can be machined to make their surfaces smoother, or there can be different slicing techniques which can be used for filling out the gaps in between these layers and making them smoother. So now we would be answering any questions or queries that you have about the whole process of 3D printing that we just demonstrated.
Thank you for the wonderful presentation. We will now be taking questions from the participants. Our first question is, which type of 3D printer is recommended for final finished products?
So for final finished products, it would be good if we use an SLA printer or SLS printer, because the materials that are being used for fabrication in these printing techniques are mainly meant for finished products such as metal and ceramics and alloys. So whichever part we make using these techniques can be used for finished products and usage in different industrial applications as well.
Thank you. The next question is, how does the printing speed affect the printing resolution?
So printing speed affects the printing resolution in some of the printing modalities such as extrusion-based printing and the inkjet-based printing. But in the laser-based printing techniques or photolithographic techniques, the printing resolution is more or less similar even if we increase the print speed and the fabrication speed. So if we have a high fabrication speed at SLA or SLS printing techniques, then the printing resolution won't be hampered, and we can increase the speed of the printing and get the parts at an earlier time.
Thank you. I hope this clarifies the question. The next question is, how can we include custom infill pattern in a model?
Custom infill patterns can be included in a model using two different kinds of techniques. One, it can be included directly in the model itself. So if you design the infill pattern and the model with the infill pattern directly, so the printer will take that pattern as a print like pattern, and it will directly print in that whatever we whatever we design as the internal structure of the object. So that we can do using the CAD designing software itself or the CAD modeling software. And another thing that can be done is a little bit like jack of all techniques. Like you can download or make different kinds of infill patterns and then overlap them onto the structure that you want to print using different softwares such as Slice3R and other open source softwares. And then the infill pattern that will be overlapped over those structures would be the infill pattern that you want to choose, and then you can convert it into a G code and then feed it to the printer. So it will finally print the infill pattern that you have downloaded or you have made custom designed for the object, and you can get your custom infill pattern through that.
Thank you very much for the interesting presentation. Now we will break for a while and then we will resume the session from 2 p.m.
Hello? Am I audible? হ্যালো, ঠিক আছে?
Only found like 6 or 5 or 6 cells. And if you compare it to the cells after incubation with a specific antibody, you can see that you have a much, much higher adhesion then. So that was promising that we could use these kind of things. So we started patterning them and incubate the macrophages also onto the pattern substrate. And again, here you can now see on the left, there are images where on the one we have M1 macrophages to a pattern that's specifically patterned to capture them. On the second part, we have an array that we had incubation with macrophages of the antibodies that capture the M2 type. And then you can see here that they fairly well correlate with the spots here. So they're specifically bound. And if you look at it when incubated with control antibody, you can again see that there is much less adhesion on these samples. And we even have, like if you look at it, then specific captures rate. So this is like the rate of the specifically captured to the one that are just adhered to a substrate like this. We have around 80%, or in the M2 type, over 80% specific capture rates here, which is very well for the macrophages here. And here you can see also a close-up, because maybe you will also wonder here why the correlation here is not as perfect as what we've seen with the CTCs. And this is again like how the macrophages behaves, and that they also still are alive and interacting with the substrate here. So if you see here one close-up of one cell that is bound to a specific point, you can see that they stick to the surface as soon as they touch such a streptavidin patch over the interaction with the antibodies that they're also carrying. But the rest of the cell body, of course, can move, and it will still try to figure out like what's happened to the other surface, but it cannot bind here, so it will not really stay always on place. So it will only bind to the part of the streptavidin. This is why you don't see them unconnected usually, but they will always connect then if they're specifically bound to one of these dots. So with this, I will come mostly to the end of the talk. So I just want to give one outlook. So as I said, in the macrophage example, you could see that one of the very interesting things is that we have coatings that are still reactive, but on the other hand also have some additional surface properties like these anti-fouling properties in particularly. And so here we have also one connection where we build up collaboration with the Mana Lab at the IIT Guwahati, So this is directly connected then to your university also. And because also he developed these kind of dip coatings that have also this favorable options that you on the one hand have like a reactive coating, but on the other hand you can also quench the reaction or you can control the wetting. And this is something that we want to develop together much further also for this kind of biological applications, because a lot of this adhesion is also coming over wetting properties of surfaces. So also here, it's just to show that we already can pattern on these surfaces, and we didn't make experiments with cells yet, but you can see here like that we have breathing patterns. So you can see the logo briefly appearing here of IITG and of INT, so for 2 institutes, when these pattern surfaces are exposed to air moisture. And yeah, in future we want to harvest these kind of wetting behaviors also for the biomedical applications. So with this, I come to the end of my talk. Here you can see our institute in the background and my group here. We see they're very international. So of course here we keep distance because it was done during Corona summer now. But still we are welcoming also these collaborations from all over the world. So I also hope to find you here in INT at some space in the future. And I thank you for your attention and of also the different sponsors here that finance the work. And finally, I want to show you the KNMF. So we also have a user facility for this kind of external collaboration. So if you want to know more, please contact me, or you can also look here on the website for the different technologies that are available. So with this, I close my talk and are here for discussion, of course.
Thank you, sir. It was really a remarkable speech. Now this is the time for question answer session. Now, someone raising his hand. I think Professor Uttam Manna raising his hand. I'd like to Professor Uttam Manna, please unmute your mic.
Yeah, thank you. Thank you, Subhendu. So, first of all, welcome, Michael.
Hi.
So, I hope things are well there, right? In Germany again, right?
It's mostly fine. I mean, we have this partial lockdown, but...
Right. So, as you said, it's a really amazing work, the way things are happening in a miniaturized way. It's really amazing. Now, so in one of your work where basically on the surface you're growing or synthesizing the small peptides, right? Mm-hmm. There are 2 different peptides. One is a flag, another is HA, right? So yes. If you see the number of sequences probably the same. Sequence is different, but the number of length of the peptide that is more or less similar. But like if you see the area of the red and the blue, right?
Yeah.
So that area is different. Why it's so? Like it's not really random. There are very few droplets with the red which are a bit smaller, but mostly the bluer smaller side and the red is bigger.
That's a good observation. So the reason here is, and that is also very interesting in the context also of your work or what I presented in the last slide, this is about the wetting properties. So as the sequences are different, also these amino acids have slightly different surface wetting properties. So after coating, because each is spotted one after the other, so after coating of a certain amino acid, then the next amino acid needs to be, let's say, kind of compatible in wetting with this. And it comes like this, that for the, I think the red is the flag one, that for this sequence, the hydrophilicity was a bit higher. So we end up with a little bit higher spot sizes. And this is another thing, like where, for example, a very well-controlled substrate, because basically, I mean, it will, if you can hinder spreading of the ink even over the initial part, then of course you will kind of control the size even better. So if we would have maybe a substrate with a certain, let's say, very low wetting properties or so, then we would maybe get even more similar dot sizes here.
Yeah, that's the place my interest. So basically, we can play with the wettability and we can really work on how to control the size and also that probably we should think about it.
And that's also, I mean, the whole thing of the synthesis one after the other on a substrate, that's very interesting. So I mean, that's really some point we can really look at then for future projects.
Right. Sounds good. Great.
Good.
And sir, thank you, sir. And the next question is from Miss Paromita. She is asking, for lithographic techniques, what is the maximum limit of viscosity for the material ink?
This is also dependent on which kind of technology. The nice thing is that we are much more flexible than inkjet printing. I mean, in inkjet printing, I'll just show this slide again. In inkjet printing, usually you will have a lot of problems with clogging of your nozzles and everything if an ink gets too viscous. Here it's mainly for the microchannel cantilever spotting. We would also need a certain kind of, let's say, maximal viscosity, but as long as the ink will flow down the channel, also as long as you can imagine of something that goes down, like, for example, a glass capillary or something, then we can also use it. So there are no limits in viscosity as long as the ink is still a fluid, let's say. What's mainly limiting us in the microchannel cantilever spotting is the drying of ink. So that means because we have this very small volumes, as I said, it's only femtoliters. So usually it's drying rather fast, also even pure water or even things that are regarded as non-volatile, let's say toluene or so, they will dry within seconds if you spot them. So what we usually do there is even to add, for example, PEG or glycerol to prevent this premature drying and be able to spot it. And another thing where you can also work with highly viscous inks that are not shown here is capillary printing. So there you really choose a small glass capillary. And there we also can deposit highly concentrated, for example, nanoparticle inks or even let's say emulsions of stuff that otherwise, especially in inkjet printing, is not printable. So I hope that answered the question a little bit. If you want to know more, you can also contact me about it, because I would always also need to look at the specific ink to say, like, could we print it directly or need some admixing of other compounds.
Thank you so much, sir. And another question is, why doesn't BSA blocking work for macrophages?
Surprisingly, we also don't exactly know why the BSA is not blocking well, because that was really our standard approach usually. Like when the people from Münster came to us and approached us about that they want this macrophage adhesion, they saw actually before us our CTC capturing publications, and this is why they said, like, okay, can we do something similar also for the macrophages? And then we did tests, because with the BSA we also started first, but it really does not make the job. Also, if you look in the paper, we also have the controls as we used like BSA, we also used treen blocking and a fish sperm blocking, and also showed like how it looks on the normal culture plate. And macrophages really like all these substrates. And we also searched in the literature, so it really, I mean, BSA reduces a little bit the adhesion, but really not as much. And so finally, we were very happy when we trialed the polymer brushes from Cesar Rodriguez group, because they were the first substrates that gave us the negative first, also where we don't have macrophages on the surface. But the direct interaction mechanism between the BSA and the macrophages, I unfortunately don't know. Also, it's also not described in literature.
Okay, thank you, sir, for addressing the queries. It was really a very interesting speech. Now, by this, we end our queries and answer session.
Thank you.
Thank you,
and
sir.
Thanks for inviting me.
Okay. Now, we'll move to our next session. Okay, so I would like to request Professor Amit Agrawal to deliver his talk on the topic development of innovative point of care micro devices. He is currently an institute chair professor in the Department of Mechanical Engineering at IIT Bombay. His research are mainly focused on microscale flow, development of novel biomicro devices, theoretical fluid mechanics, and turbulent flow. He has published more than 200 journals and a book. Also, he has filed a dozen patents with his students. His primary contributions are in the development of a unique blood plasma separation microdevice and derivations of equation which he believed are more general than Navier-Stokes equation. Also, he is the editor of 3 reputed journals like Experimental, Thermal and Fluid Sciences, Nature Scientific Reports, and Transaction of the INAE, and also elected Fellow of Indian National Academy of Engineering and National Academy of Science, India. He has been awarded with many prestigious awards such as the Development of Atomic Energy, Department of Atomic Energy DAESRC Outstanding Investigator Award, Professor K.N. Sitaramu Medal by Indian Society of Heat and Mass Transfer, Professor H.H. Mathur Award for Excellence in Applied Science by IIT Bombay, and the Shanti Swarup Bhatnagar Prize for his outstanding works. So I like to request Professor Amit Agrawal to please move forward with his speech now. Please, sir.
Good afternoon. So thank you for this kind invitation. Grateful to Professor Bandal and Professor Manna for having me here. So I think I can start presenting my slides here. Let me do that. So can you please confirm that you can see my slides?
Yeah, it's visible. Yeah.
Okay, thank you. So, so once again, thanks to the organizers for having me here. So I'm going to talk about some of the development work in biomicro devices that we have done over the past few years. And of course, this work has been done by a lot of students and over a substantial period of time. And I would like to really acknowledge all of my students who have contributed to this effort. So let me just start off by saying, I'm sure as part of this workshop, you are already getting exposed to a lot of such devices, but just to set the stage, I would like to sort of say that there are a lot of small devices, handheld devices that are becoming very popular, like what is shown here in this picture, which are useful for monitoring the human health. And of course, people are looking, so these can be like glucose meter, or they could be like a pregnancy kit. So these are all small kits. And of course, there is hope and interest in developing more and more of these. Of course, and the requirements would be that they should be simple to use, they should be cheap, they should give us quick results, and they should require a very small amount of sample for their processing. So of course, these are all the requirements, but how do we go about making them is what we have to sort of look at as an engineer. So this is just one of these devices. If I just look inside one of these devices, what you would see is there are several modules, because we'll have to make things, make various modules, then combine them together. So there are, like this is a sensor which was used to make dopamine, detect dopamine from whole blood. And you can see that there are mainly two modules. One module is for the sensor, and the other one is for the sample preparation. So you can see the two modules on the left and the right side of the figures. So one is where you put in blood, which comes in, and you do certain things to the, and you get maybe the plasma out. And of course, this is the device which was built by us in our lab. They were using it. And there is another sensor, another module, which is a sensor module, where they are having all these, I mean, some sort of a sensing elements, which would sense whether the dopamine is there in this blood plasma and the plasma of this blood or not. So this is just to sort of give you a sense of what goes on into this, inside what is inside the black box that you are seeing in this picture. Of course, there can be more sophisticated electronics and readouts, etc., maybe powered by a battery and so on. But I mean, essentially, we would have these modules. And what we will be concentrating on here in my talk is to talk about this first module, which is a sample preparation module. So what it means is that you take the sample, it could be blood in this case, it could be saliva, it could be urine, it could be anything else, any other body fluid. Of course, blood is the most common one. So you would like to take that sample from the patient and then process it in some way so that you are able to detect, or you are able to bring it to a form where then you can detect the quantity of interest, like what is done here by taking the sample to your next module, the sensor module. So these two modules are, you can say, are always going to be there in any micro device. So what we want to talk about with this as a platform or the background, let us say that what we will talk about are modules like what we showed you in the previous slide for the sample preparation. So there can be several steps that may be required. Maybe certain things have to be, it may be either separating certain things, or it could be adding and mixing things. It could be heating things. So those are the type of common operations that are done. And again, as engineers, we would have to devise things so that one is able to do these things effectively. So what we are going to talk about here are the following. We will talk about if we have to extract plasma from blood, how do we go about doing that? And so in that regard, in that context, we will talk about the blood plasma separation micro device. We could also think of extracting not just plasma, but also have platelets, which are, should be rich in platelets, selectively rich in platelets. And so then we can have a platelet-rich plasma micro device. We will also see that there is a need to focus cells. We will see the examples of that later on. So again, another device that we will look at is to focus. How do we focus these cells selectively? Or sometimes we have to heat the samples or to maintain the temperature of the sample at a certain temperature. So then we will talk about the constant wall temperature micro device. So these are the various micro devices about which we will talk about. So you can see that there is a lot of material that I have. Of course, if you have any questions in between, feel free to stop me and ask me. Or if you want, we can just wait and talk about these at the end. And of course, if I'm going over time, please stop me at any stage. Now, so what we will talk about here is how do we go about designing these modules. We will see what parameters, there are a lot of parameters that would affect the performance of these devices. So we have to understand what are those parameters, how do they affect, whether it is the geometry which is affecting, whether it is the flow parameters like flow rate, etc., or the some other flow properties which are important for us. There could be also fluid properties, for example, the viscosity of the fluid, which itself may depend on the type and the percentage of cells that is being carried by your fluid. So all these things would start mattering, and we will start to see how to design these things, keeping these parameters in mind. And of course, the objective of this slide is to just show that there are a large number of these parameters which could affect the performance of the micro device, and therefore we'll have to slowly optimize things and by understanding how each of these parameters is affecting my performance. So we will start off with our first micro device, which is our blood plasma separation device. And the need for this is very obvious because blood test is one of the most common tests which a doctor prescribes to a patient when the person is sick. And this is because, so what is done is blood test. In blood test, the blood is drawn from the patient, maybe a few milliliters of blood in a syringe, and then it is centrifuged to separate out the plasma. So if this is just to give you a mental picture, if you centrifuge the blood, then there will be plasma, the liquid part of blood, which is of course the carrier part when the blood is flowing in your blood vessels. And then the cells.
Hi, this is Paul Beckman with DSP Concepts. We're a third party that worked with ARM on the development that's included
would sink or sediment
here.
at the bottom.
It in MATLAB. Pass it generated by MATLAB.
They will centrifuge.
Time domain response.
At the bottom of your test
and
tube.
In this point right here, in a 15 kilohertz component
here.
in this example, find it.
Then you will have white blood cells sitting on top of these. So, and this plasma,
here,
which is the liquid part, has
pointers to where the unique F state variables,
which are of,
and we also give it a block size, how many samples
body.
will be processed.
So, as we were
per
talking about,
function.
when a blood test is done,
next
the
we loop
blood
over,
is withdrawn
write
from
the output data here
of the patient, and then we can we have to to separate out the plasma, look at these biomarkers using the type of sensors that I was showing you in one of the earlier slides, and then. So you can see that
Maybe
the plasma separation from blood is a very, very important step, and this is going to be required for irrespective of what type of biomarkers you're looking for in your bloodstream. So whatever disease you're trying to detect, the corresponding biomarker you will have to find in your plasma, and therefore separating out plasma from blood is a very, very important step. And because we are going to separate out plasma from the remaining constituents, we have to understand what is the proportion, what is the size, so that we can devise our strategy to keep while keeping in mind the number density or the number of the cells which we have to remove, their sizes, and so on, the shape maybe. So what we see here is there are three major constituents of blood. Of course, four, maybe the three in terms of solid, which are red blood cells, white blood cells, and platelets. So in terms of volume, the RBCs constitute around 45% by volume, and this is because there are very large number of these RBCs, around 5 million of these RBCs per microliter. So just imagine that if you have a drop of blood, which is several microliters, then you are having maybe 30-40 millions of these RBCs in there. Then there may be, in terms of WBCs and platelets, they are not very much in terms of their volume that they would occupy upon centrifugation. WBCs are relatively larger in size, but smaller in number. Platelets are smaller in size, but in a relative sense, with respect to WBCs, they are slightly larger. But with respect to RBCs, you can see that the proportion of WBC and platelets are quite small. So, so when we say plasma, we are going to basically be looking at trying to separate out RBCs because that's the most important component. And if you can remove that, then hopefully we would have good quality plasma. or very relatively free of cells that we would have. So as we were talking about, the conventional method of separating out plasma is to take it to a centrifuge and just centrifuge the blood that you have collected in your test tube. And of course, as we have seen in the previous slide also, there would be separate layers that would form. However, the issues with this method is, of course, sorry, at the very beginning we said that we would like to have a small handheld micro device, and you can see that this centrifuge is not going to be integrable with that sort of a small micro device. You can also see that this is going to require a lot amount of energy, and of course you can see that typically a lot of samples are loaded in there. So multiple sample means that maybe there is a possibility of mislabeling and therefore prone to errors. Of course, so I mean, you can imagine that definitely the centrifuge is not integrable with your micro device the way that I mean, people are envisaging at this stage. So therefore, we need to move away from this centrifuge and be able to do what this centrifuge is doing in terms of plasma separation in a micro device itself. And it just turns out that it is not a very straightforward problem to deal with. But before that, let me just sort of show you that if we could do that, then in a small device like what we are ultimately able to construct, and I'm going to talk about more, but this is just an overview of what we would like to get to, is that we would like to have a very small, like a coin shape, a coin size sort of a device, which has all these nice properties in terms of a small amount of sample that it would require. It is very portable, as you can see. It can be used anywhere, including at the site of the bedside of the patient, and so on. So these are all the advantages of point of care micro devices that I'm sure you know about. And of course, we would like to give that functionality, all those functionality to this small device here. So how do we go about doing that? So as we said that this separation turns out to be not a trivial problem. A lot of different groups worldwide have tried to solve this. One obvious sort of a solution which comes that if we sort of think about that we are looking at solid, trying to separate out solid from liquid. So the natural choice is, can we just not use a filter? So we can use a filter, but the issue is the numbers that I showed you in one of the tables here. So we said that we have, so if imagine that if you have to process 1 milliliter of blood, then you are dealing with 5 billion of these cells. Okay, not million, it will become billion because 1,000 times you have increased the volume. So, so if you are dealing with a few milliliters, let us say 3, 4, 5 milliliters, then you are talking about several billions of these cells, and these cells will come and clog your filter. So that is one obvious thing, obvious problem that you see. So first layer, as you can see, the layer by layer, the cells will come. They will start accumulating on the filter surface. Some of them, RBCs, are also very deformable. So some of them will actually pass through these small passages, much smaller than their size. So initially what you will see is that these cells are able to pass through, and you are not getting very high quality plasma. Later on, what you would see is that because several layers by now have formed while a few of them are passing through, so because of these several layers which have formed on this side of the filter, the further flow of blood or plasma from the left hand side to right hand side is essentially stopped, and that's what we mean by clogging of the filter has happened. So that's a typical scenario. And that means that the filter is no longer doing the job. Whatever plasma you could get is actually quite contaminated, and it is very small in quantity. That is what we're getting in this process. And of course, the other thing you can easily imagine, as we said, that we are going to ultimately look for these biomarkers in plasma.
Yeah, in one of my previous videos,
you can see
I was
as
already
the
talking about
biomarkers
IR filters,
are
which I have
passing through
implemented on the
this filter
STM32.
or this
But now let's
membrane
have
that
a
you are
look once
having,
again what
and this
exactly an
membrane
IR
is going to
filter
have very
is
large
from the
surface
structure
area.
point of view.
So the biomarker can actually get deposited on the surface of this membrane and not come out on the other side. So that means, or even if it comes out, it is coming out with a reduced concentration. So that means the sensitivity of any test is expected to now reduce. So you can see that although filtration is a very common sort of solution which comes in mind, it is not a very good solution. And of course, so that's why initially you will see that these sort of works were done in 2005 and 6, but later on people realized and they have started to look for alternate strategies, something like what is shown here. So instead of having a direct filtration, you say that, okay, I'm now going to have sort of an indirect filtration. So I'm going to let the flow happen. And then laterally, there will be some plasma or some liquid which will ooze out. And what we expect is, again, the cells, because of these barriers that we have created, very few of these cells will go to the other side of this, which is the stream which is coming from here. So essentially, there are two such bends on the two sides and separated by these pillar-shaped barriers. And so most of these cells are essentially in this bloodstream, and some plasma is oozing out between these pillars, and they're getting collected somewhere here. So that is the idea, and an idea tried not too far back. And of course, this is still, you can see it is still like very much like a filter, maybe not a head-on sort of a filtration it is doing, but it is doing something very similar. At least it is a mechanical blockage that you are creating. And the disadvantages that we talked about in terms of the large surface area and the biomarker getting absorbed on the surface of this filter still remains even with this design. So of course, you can see here, or even other cases, you can see that there are a lot of such marbles, for example, one can put in, and then one can try to get the plasma coming out. But again, you can see that this is very similar to like a filtration-based device. Some small variations here and there, but by and large, the same idea. So of course, there are several other class of devices that we can look at, but we are not going to sort of look at everything here. We are just going to sort of now come to the class of devices that that we are going to use because of their promise, which is like devices like what is shown here, where what you do or what you rely on, you know that when the flow is happening, there are certain hydrodynamic forces that comes into the picture. So for example, what it means is that if you put an object in the flow, so for example, if I put a ball in the flow, and if I'm flowing the air with a very large speed, then this ball may have a tendency to lift, just like an aircraft would lift up. Okay, of course, this would require a certain set of conditions to occur, but I can generate certain forces because of the motion which is there. So those forces and those motions, there could be a drag force, there could be a lift force. So those forces I'm going to utilize for the separation. So I'm going to sort of say that as the cells are flowing, they should have, or they would experience a certain amount of force, which of course I have to study what type of forces come in, what direction will the force be, and so on. So, but once I understand what forces will come in, maybe I can have a bifurcation, or I can have a separate passage for these cells to pass through, and I would have a separate passage for the remaining plasma to have. So that is the basic philosophy around which this would work. So these are all hydrodynamic forces based designs, which we are saying is because of the hydrodynamic forces which arises because of the motion of the fluid itself. So this is the type of sort of philosophy which we would adapt and use for our separation purposes. So of course, what you see here in this slide is something which was taken from the literature. And of course, you will see that they have very intricate features that are there. Something which is 20 microns, you can see that this is a very complex shape that has been given. And these shapes means, so you have to also understand that when we are using these devices are mostly use and throw. You are only going to use it for once, And then to avoid any cross-contamination, you don't want to use it again. So if you have such a complex design, which makes it very difficult to also manufacture, so then you can see that I'm not going to, I mean, making all these intricate features means that manufacturing cost is going to be reasonably high. And that means that it is not such a good way to just use it once and throw it off. So, and of course, making this means that even if I have small issues with the dimensions, I would have, I mean, I could have problems, or I could have deterioration in the performance of the device. So these are some of the issues that one can identify with these existing devices, that they have very small features, sizes, as we saw, 20 microns and all, which means that they're very difficult to make. Any small, like just imagine that 1 micron difference is what I'm getting in my dimensions, and 20 becomes 21, or 20 becomes 19. So it becomes 5% change in your passage cross section. And that means that's a huge change in terms of the performance that one could expect. So that's the type of issues that one see here. And of course, it also means that they are very prone to clogging. We have seen that the RBCs are something like 8 microns in size, and this passage size is only 20 microns. So that means 3 cells coming together is enough to clog these devices. So again, you can see that, and this is not very unusual because we're talking about millions and billions of these cells. So 3 of them coming and clogging is very, very probable. So it's very, very, I mean, just flowing together, if they're close together, if they sort of start flowing as a clump, 3 such cells, then they could clog your device. So you can see that these devices becomes practically very difficult to implement because of the issues that we're talking about. And that's why a lot of these devices were actually demonstrated on diluted blood. So you essentially artificially reduce the number of cells that are there in your bloodstream or in your sample stream, and then your device would work much better. The other issue, so this is one set of issues that one can find. The other set of issues that one can see is that they have very low rate of plasma extraction. So that means that to get any meaningful amount of plasma coming out from the other side, I will have to wait for half an hour, 1 hour sort of a timeline, which is of course very, very large and something which we cannot necessarily afford. So the question arises is, can we simplify these designs? So, so with this in mind, so this forms the background around which we are going to now talk about and look at how we can make a device which is better than them, which do not have these issues that we have talked about, and it is still simple to make and cost effective and so on. So we will now talk about a few slides about the science, about how one can make these, what are the type of effects that could that comes in when the blood flow happens in the small passages, and how could we exploit these effects or these physical phenomena that exist for making a blood plasma separation device. So there are 2 pictures which are shown here in this slide. One is a schematic, and the other one is an actual picture which is taken from our lab, where what we see here is a simple bifurcation sort of a thing, or a T sort of a thing. So a T-shaped channel. So we are pushing in blood from one side, from the left here, and of course that these 2 are the 2 outlets. And what you see here is, interestingly, so what is schematically shown is that most of the cells are going towards the right, and very few of them are going through this side line or side branch. And this is because, and of course you would see that in practice also happening. And this is what is known as, this is happening because of the so-called the Farias effect, where as these cells move in a small passage, they tend to move away from the walls. And this happens because at the walls, the shear stress or the shearing is maximum, and the cells have a tendency to avoid regions of high shear. So you can see that velocity gradient, which is proportional to this shear, is actually minimum at the center line. So they will tend to move towards the center rather than moving towards, and that's why you would have a cell-free region which is going to be formed near the walls. So again, that is what has been shown here schematically, and this is also what you see. So what you see in your right figure is you have these black lines, which are indication of the walls of the microchannel, and then you have a light colored region, like sort of a whitish region, which is an indication that there are no cells in this region. And then in the center of the cell, you have these red or dark colored region, which is dark, which is essentially indication of that this is where the RBCs are now. So what we are talking about is the cell-free region is also now what is being formed. And then you can actually take out, because the cell-free region are forming on the wall itself, you can try to ooze out, slightly remove this plasma, because on the walls is all that you have is plasma. So if you are careful, you can actually get some plasma out. And of course, this is using this various effect that we are utilizing in this case. The other effect that you could look at, you can utilize, is the so-called the bifurcation law, where we can bring in the blood through a bifurcation. So now you have, once the cell comes at this bifurcation, it has a choice to either flow in this upper branch or in this lower branch. And what has been found, and that is why this name was given, is that the cells will prefer to flow in the branch which has a higher flow rate. So if you maintain the flow rate at least of the in the branch which has a higher flow rate to be at least 2.5 times of the flow rate in the lower branch, and you maintain the size ratio of the cell to the passage size of the order 1, then you will start seeing this effect kicking in. And this is something that you could utilize, because this is a very interesting idea and an interesting thing that you could utilize to make a device. And of course, this sort of happens because once the cell comes at the junction, there is a certain amount of pressure distribution, and this pressure distribution leads to a force, and this force tends to push the cell in a certain branch, or the branch which has a higher flow rate. So using these sort of effects, one can make a very simple device like what is shown here, like a teach microchannel based device, and one can get good amount of plasma here. And but the only catch here is now we have the blood is extremely diluted. So just like what we had seen from the literature survey, people have demonstrated simple devices which are diluted, and we can see that very simple device like this can work very well if it is diluted enough number of times. But we are not interested in that. We are interested in being able to work with undiluted blood. So we'll have to now look further and bring in additional effects, additional things in there. So what is being done here, what is being shown here, is that you can take a channel and you can pinch it. So that means that you suddenly reduce the cross section of the channel and then you expand it out again. Now, when you have this certain contraction and then certain expansion of the passage happening, especially when you look at the certain, so when the contraction happens, of course, the flow velocity will tend to increase. We know that as the flow goes through a narrower passage, the velocity tends to increase because it has to conserve mass. And then when we come on this side, because the velocity has been increased now, the inertia of the cells becomes very different from the inertia of the plasma. And therefore, when we suddenly expand them out and the velocity is suddenly reduced, we will find that they have their own trajectories, and the plasma will essentially come in and sit in this corner, and the cells will tend to move straight and follow a passage like what is shown here in this dark region. So you can see that there will be a corner region where the plasma will get separated out, and this is from where you can ooze out, or you can take out the plasma. So you can bring about, you can utilize this effect. Of course, these are all partial effects is what I would say, because you will see at the end, we'll have to put all these things together because all of them would work reasonably well with diluted blood, but none of them works very well with whole blood. So you'll have to somehow combine all these things together in a useful manner. So here what you will see is another sort of an idea which is being shown here, is that if you take this blood and pass it through a bent passage, then because of the centrifugal forces that would arise, the cells will tend to move towards the outer side of this curve of the bend, and the inner side is where will be cell free, and you can take out the cells, the plasma from this side. So now what we can so with all these things effect sort of understood, what we are going to sort of show now is that one can make a device like what is shown here, where all these effects, whether it is a bifurcation that you are seeing here or you are seeing a bend here, so all these effects are now being brought in. Of course, you can see that there's a change in area between here, and then there is a change in area again. So all these effects have been put in together to be able to make a device like what you should see here. And of course, you can do you can collect the sample which is coming out, and then you can do a detailed analysis using flow cytometry. And you will see that this region, which is an indication of RBCs, is essentially sort of missing or at least substantially reduced, which is an indication that you have removed a large percentage of these RBCs. And of course, there is a lot of quantification of these results that has been done, and which clearly goes on to demonstrate that we are indeed separating out the plasma or the RBCs and getting good quality plasma from our device. So this is a slide sort of summarizes what are the unique things that this device can do. It can achieve almost 100% separation efficiency with whole blood without having to dilute the blood. You can still get 100% separation efficiency, and you can get process it very fast. 10 ml or 5 ml of blood you can process in 10 minutes if you want. These are all utilizing very large dimensions, 100 microns and larger. So then there are no problems of clogging, dimensional tolerances, etc. is not an issue. And of course, we have also done detailed characterization of the plasma that is coming out, and they are hemolysis free and so on, and no loss in proteins and so on. So that means that this is good quality plasma that we're getting. So it looks like we are going very slow here. So I will just very quickly talk about one more device and then we'll wrap up. So we will see that we can extend this idea to be able to get not just plasma, but also platelet-rich plasma. And of course, as we know that platelets are very important. If you have, for clotting and so on, they help for the blood to coagulate. And so what we can, and of course, these platelets are going to be very important when you are trying to do a treatment for dengue and certain surgery and so on. So platelets are very important, and having platelet-rich plasma is going to be required in a lot of these applications which are given here. So with this sort of a background that we saw that we have in the first part, we could devise a simple device like what is shown here. And to show that, again, using flow cytometry, one can see that this part now, which is an indication of the platelet region, has been substantially enriched with respect to whatever is incoming sample. And if you were to quantify this, and of course, this is how things would look like if you were to collect it. And if you were to compare it in terms of fold enrichment, in terms of how many platelets you have with respect to whatever platelets you were putting in terms of the ratio, then you would find that it has become 15 times more in this device. And of course, there are several designs that we were testing. So at least we can have easily demonstrate 9 to 15 times of enhancement in terms of platelets that we are getting. And the clinical requirement is typically 3 to 8 times. So this is well above that clinical requirement. And we also can very quickly test whether these platelets are getting activated or not, because that's one sort of an indicator of how good a quality of platelets you're collecting. So this is a platelet activation test that was done. And what you see here is that the inlet sample, which is the green thing here, versus if you were to centrifuge this sample. This is a conventional method of preparing your platelet-rich plasma. So you can see that with respect to the green curve, the red curve has actually shifted a lot to the right, which indicates that all these cells have actually gotten activated. So approximately 50% of the platelets get activated in the centrifugation process. This is a known sort of a thing and also shown being demonstrated from our results here. And if you were to pass it through our device, you see that, which is the blue curve, you will get the blue curve. And you see that they are almost, the blue and the green are virtually overlapping, and there's a very small amount of activation, less than 6% activation that happens in our case. So this is just an indication, and we have done other sort of tests to sort of show that our device is actually doing quite well. So I don't think I have time here, so I will just go towards the sort of the end of my talk here and just sort of summarize and say that what we have talked about is the blood plasma separation device, which eliminates the need of the use of a centrifugation altogether. And this is something that we could demonstrate on whole blood. And it could also result in a huge saving in terms of time, energy, and cost. Now, of course, there are other devices which we could not talk about here. So let me just stop here and thank my students and other people in the lab who have substantially contributed to this device. So it's essentially Siddharth Tripathi was instrumental in forming or making this blood plasma separation device, whereas Vijay Lakshmi was the one who actually made this platelet-rich plasma separation device. And of course, I would like to acknowledge all my funding agencies and all the collaborators. And I would like to thank you. And I would be happy to take any questions here.
Thank you so much, sir, for your excellent speech. Now we have a question from a participant. She is asking, won't pulling out the plasma from the cell-free region of the channel also in turn attracted the cell to that region because of the force used?
Yeah, so as I said that there are several forces which are there. For example, in this case, there is a shear, so there's a shear induced migration or movement of the cells that you can see is what is getting important, what is kicking in here. So that means, as we were trying to explain, that if you were to look at the velocity distribution in the channel, you will see that the blood velocity is 0 at the walls because the wall is stationary. The flow at the wall would be stationary. And then from the wall at 0, it will increase to a maximum value at the center line. And then it will go again to 0 at the other wall. So it goes from 0 to a maximum value and then goes to 0. It follows a parabolic profile. Now, if you were to take from here the velocity derivative, of this, which is the velocity gradient, you'll find that it is essentially it is 0 at the wall. So it is maximum at the two walls and it is 0 at the center line. So that means that the shear is very large at the walls, and the cells, as we said, wants to move away from these regions of high shear. So that is what is causing this movement of the cells from the walls towards the center line. So I don't know if that answers your question.
Thank you, sir, for your speech, for addressing the queries. And sir, if anyone asks anything, then you can just raise your hand or post the question in the chat box, please. Thank you, sir. I think there is no other question.
Okay,
thank you very much, sir.
So I will log off then in that case. Is that okay?
Okay, sir.
Okay, thank you then. Bye, everybody.
Yeah, in one of my previous videos, I was already talking about IRR filters, which I have implemented on the STM32. But now let's have a look once again what exactly an IRR filter is from structure point of view.
Hello.
You can see this here on
Yes, sir.
Wikipedia. We have like
now
a
we'll
couple
move
of
to our
delays
next
and we have a couple
speaker.
of multiple. Yeah. So now let's
should I go ahead and speak? This is Dhananjay here.
Sir,
so now let me give
moving on, it will be my honor to invite Dr. Dhananjay Dendukuri to deliver his speech. Currently, he is a CEO and co-founder of Achira Labs. He received his PhD in chemical engineering from MIT in 2007, where his thesis work received the Centuria Prize from the best dissertation in the area of micro and nanotechnology at MIT in 2008. Prior to that, he obtained a MASc in chemical engineering from the University of Toronto in 2002. During his BTech, he also received Institute Blue from IIT Madras in 1999 for his outstanding all-round performance. His work at Achira Lab was recently recognized through his being named as one of the 20 individuals to receive the MIT's Technology Review's prestigious TR35 award in India. Also, he has many papers under his name. So I like to request Dr. Dhananjay Dendukuri to please take over and deliver his speech. Please, sir.
Okay. Can you all hear me?
Yes, sir. We can hear you.
And can you see my screen?
No, sir.
You can't, is it?
Yes, sir.
Okay, 1 second. So now? Can you see the screen now?
Yes, sir.
Okay, great. All right. Thank you, everybody. And thank you, first of all, to Dr. Biman Mandal for inviting me to give this talk. And I've had many interesting interactions with other faculty at IIT Guwahati also recently, including Dr. Deepankar. Unfortunately, can't be there in person. Would have loved to meet you all, but I guess this will have to be a substitute till we meet in person. So today's talk is going to be a little bit about the journey of a point-of-care diagnostics company to take a product from R&D into market. So hopefully give you people a little bit of perspective on that and what does it take to make a product ready to get into market. Okay. So this is a little bit about Achira. We are a point-of-care technology company based out of Bangalore. We're an ISO 13485 certified company with licenses from Government of India to manufacture diagnostic products. Our core actually is that we are an R&D company focused on point-of-care technologies, and our main platform is based around a hydrogel sensor technology that is embedded inside a microfluidic device. So my talk predominantly will feature that today. The platform itself is covered by multiple peer-reviewed publications as well as several granted patents. The company is founded by myself, Dhananjay, and also Suri, who's also a PhD from IISc. We've received generous support over the years from many organizations, including BIRAC, which is, of course, as many of you know, under DBT, and also some support from ICMR as well as Grand Challenges Canada. And we've raised private money from Catamaran, which is the family office of Mr. Narayan Murthy. So that's a little bit about the background of the company. And because everybody wants to know what is your company doing during COVID, I thought I'll actually start with that. Because every diagnostics or healthcare company has to do something around COVID to survive. And we are no different. So I thought I will start the talk by just telling you a little bit about what we're doing around COVID. Before I get into the point of care. So we're doing actually several different things. We have actually developed with BIRAC support again, an ELISA test. So an ELISA is a test that actually this test will actually pick up antibodies against COVID antigens. And especially as the vaccine is coming along in the next few months, we're all hearing, it will be very important to judge the immune response of different individuals who get vaccinated. And usually you expect that people who have a good response to the vaccine will generate IgG antibodies against the spike protein of the virus. So we have a test that is actually going through approval right now under ICMR that will be able to measure these IgG assays. Secondly, we are also manufacturing and marketing an RT-PCR test for COVID. This is actually being done in collaboration with a company called GenePath out of Pune. So they have developed this test and Achira is manufacturing it and also marketing it. So this is something that the last 6 months has been sort of a turbo learning experience for many companies like ourselves in terms of learning super fast the entire regulatory process and what it takes to scale up manufacturing and so on. So happy to say that we have in fact now close to 1 lakh plus samples have been done on the kits that we have sold. And as you all probably aware, a lot of RT-PCR tests is being done. Predominantly, a lot of this was with international companies early on. Now there are several Indian companies, but we believe we have an excellent product. And we're also developing some antigen assays for COVID in the and so now I will actually switch to the main topic of the talk today, which is point of care diagnostics. And in order to understand point of care, I think it's important to understand the lab market structure overall. So on top of this lab tree, you have these reference labs, which are very large automated labs, like ThyroCare or LAS, and you'll hear many of these big lab names, and they have large, hundreds of thousands of square feet of highly automated labs, okay? And this is, you could say, on top of the tree in some sense. And this is very close to what happens in the Western context also. But below that, you have these satellite labs, which have smaller, semi-automated, some pieces of automated equipment. And then even below that, you have collection centers, which are smaller labs or even doctors' offices, which actually generate the samples that eventually go up this tree all the way to the top. So the focus of a platform like ours is actually at the bottom here. That is, we're talking about the very large number of smaller doctors' offices, collection centers, etc., and not these very automated reference labs. So why is it important to do point of care testing, right? Because one question that you could ask is, well, there is already these highly automated, reference labs, and is there really a need to do point of care testing? And I think actually this question before, we had to do a lot of explanation, but now I think COVID has sort of self-answered this question to everybody, which is that it's fairly obvious to everyone, especially in an infectious disease context like COVID, that if you could do testing near the patient, if you could do testing in the field, if you could do testing in a mall, if you could do testing in a railway station, there are very obvious advantages in terms of, just easing all of our lives, right? So this question has got answered in itself to the public, but nevertheless, there are a few other points which are important to make. I think, especially in the Indian context, a couple of the ones I've circled here. One is that you have a lot of inaccurate results, potential for inaccurate results, because samples are not stored properly and you have degradation by the time it reaches a lab. And you also sometimes have this issue of patient compliance. That is, if you are asked to do a test, and many people actually don't do the test. Again, I'm talking about non-COVID context here. And you don't have compliance. So being able to, in a one loop, actually prescribe a test and do it has certain obvious advantages. And I think these are only going to grow with time. And the central lab, I think, will never go away. It's always going to be the reference standard, because there's a certain amount of quality and economies of scale that will apply in a reference or central lab setting. Not to mention the fact that there's a very long list of tests that need to be performed, not all of which can be performed at a smaller setting. However, I think there is a compelling case for many common tests to be performed in these distributed settings. So we have focused on immunoassays for this A6 platform. So why immunoassays? So the answer really is a commercial business reason that many of the point of care settings already have semi-automated chemistry analyzers that are available, many from Indian companies and already at quite a low cost. However, immunoassay solutions, immunoassays are tests for proteins basically, and these can be across clinical areas like I've shown here, which could be endocrine or fertility or fevers. So you have immunoassays across clinical areas, but usually you have lower numbers on these tests, even though the costs are higher. And many of the commercial solutions that are available today are made for higher throughput settings, where you have at least dozens or hundreds of samples a day. So there's a sort of a business logic here which prevents immunoassays from being done at these lower throughput settings. So this is exactly the kind of market that we are looking at. So what is needed in terms of the characteristics of a test? So these are some of the things that are important, and also based on our own experience looking at the field. I think one is you've got to have a test which is easy to use. And what that means specifically, for example, in the Indian context is you don't even want something like a time step, right? So often if you do these assay procedures, it will say something like do step A at the start, and then wait for 2 minutes and then do step B, right? So you don't want things like that, because those are exactly the kind of things that people can go wrong with or forget to do, etc. You also ideally want to avoid pipetting steps. So people may not calibrate their pipettes, people may not have the right pipettes, etc. So you want all your reagents to be the cartridge should contain everything. So that's important. You also want, again, room temperature stable reagents ideally, both to cover shipping, because India is a country with very high range of temperatures, all the way from negative to 45 degrees. So yeah, sorry, I'm back. Are you still able to see my presentation?
Yes, sir.
Okay, sorry, I just lost connection for a moment. Okay, so the consumables have to be inexpensive, and for a new technology, this is often a significant challenge, because when you talk about a microfluidic cartridge with molded and assembled with all the reagents and so on, to be under 100 rupees is not an easy thing to do. And to actually the next thing is, do you want rapid results? So under 15 minutes for a test result, that's about the time people are willing to wait. And yet all this should be done with the same quality as the reference lab, right? And this is quite challenging, because reference lab procedures have a lot of automation and a lot of quality controls built in. So in order to say that you are the same quality as the reference lab actually is a pretty challenging mountain to climb. And you want to do this ideally with capillary blood samples, because there may not always be a phlebotomist who can collect blood for you. So these are just some of the requirements of a good ideal test. So this is our platform. It's a combination of a reader and a cartridge shown here, which we call ASICs. So we have one version of it, which is 100, and then we are about to kind of come out with a newer version, which is called the ASICs 200 in the coming year. But just to go give you a little bit of context about how these kinds of things actually can come from an academic setting. So my PhD work, in some sense, flowed into this and kind of led to the creation of this technology. So this technology that was invented during my PhD is called flow lithography. And basically, flow lithography is a way of making microparticles in flowing streams of polymer. So this is what is shown here in this video, where you can see this M, I, and T letters are being formed by photolithography. But unlike standard photolithography, where when something forms, it stays stationary, in this case, they are being formed and they're also able to move. So there's a very clever trick around the oxygen inhibition, which enables a lubrication layer to be present around these particles so that you can actually flow them. But the idea here is that you can use a mask, a photo mask. So any of these For example, if you have a photomask, you'll get this. If you have a square, you'll get a cuboid, etc. So you can make really any two-dimensionally extruded shape with this flow lithography. And the nice thing about this is that a lot of bio-friendly polymers, like PEG-based polymers, for example, are not amenable to standard lithographic techniques like spin coating and curing and so on, because they're not very viscous, correct? However, if you can actually flow such a material in a microchannel, you can then subject it to photolithography like this and form now sort of these really bio-friendly particles. And there are several applications that now people have found since I finished my PhD. A number of companies have been started around this technology, but we are using them for the fact that you can embed, antibodies or oligonucleotides or other sensitive chemicals, biochemicals, into these particles and have them polymerized inside, so that they become sensors. So this is really a way to make microparticle sensors, basically. So then what we do is actually these sensors are embedded inside a micro cartridge shown here. This is just a cartoon view. It's not the exact thing, but the basic point is that those sensors are made in large numbers, by tens of thousands even, and then embedded inside a cartridge. And then you have a sequential flow of sample and detection antibody followed by a wash. These are fairly standard steps from the biochemistry side, which leads to ultimately a fluorescent signal emanating from each of these sensors. And so the capture reagents are actually stored inside here. The sample contains antigen, and the detection antibody is a secondary reagent, which forms what's called a sandwich complex on each of these sensors. And by simply putting different antibodies in different spots, you can form a small array and do multiplexed panels. So you can have 3 different tests on a cartridge, for example. We've done even up to 8 or 9 different tests using this kind of an approach. So this is what a cartridge and the sensors look like in real life. So you can see these tiny white spots here, and if you zoom in, you'll see those sensors embedded inside. And we have these adapters which actually contain all of the reagents and the wash buffer and so on, so that the customer is presented with a fully loaded cartridge. So this is some data that we've actually published on this platform, where you get very good quality data compared to, I would say, gold standard immunoassay platform. So this is just a calibration curve showing that as you increase the concentration of some hormone, in this case, this is HCG, which is a pregnancy hormone, you get a very linear increase in fluorescence, which is plotted here. And the nice thing about this is these hydrogels are 3-dimensionally porous, which means that you don't get just a surface signal, you get a 3D signal, and you're able to get molecules to diffuse all the way in and give a very strong signal. So that means you can get very good, strong fluorescence sensitivity, even at low concentrations and low times, which is obviously very important. And as one example of that, we're able to do a cytokine test for something called IL-6. In fact, IL-6, by the way, is a very important test for COVID right now, because it's been shown that these cytokines correlate with patients who get really badly sick. So, and something like an IL-6, you could do in a point of care format on our platform. And that needs like picogram per ml type of sensitivity, which is not easy to achieve. The other thing you can see in these images is just photographs showing as you go from 0, which is kind of dark, all the way to 250, you can see with your eyes that you have increasing but very uniform amounts of fluorescence from our sensors. And that fluorescence is actually captured and quantified on these calibration curves. The other thing, we keep publishing work in collaboration with academics, and which is to kind of stay true to our R&D roots. So one of the things that we found, which is quite interesting, is that these particles actually, they have, even though they're porous, there is some barrier, because there is the porosity is not, you have a reaction diffusion boundary layer effectively that is shown here, so that there is a constant competition between things that are trying to diffuse in and the fact that they are reacting and getting bound there. So that defines a certain boundary layer here. And we found that that edge fluorescence actually can be used. If you pick up just the edge, you get a much more enhanced lower limit of detection by about a factor of 5. We are able to improve our limit of detection from about 0.3 or so to 0.05. by changing the shape of these particles and actually utilizing this edge effect. So you have like a wheel-shaped particle shown here as opposed to a disk. So then all of the signal is very bright. And so that actually can enable you to bring down your lower limits of detection. In terms of the assay workflow, this is what we do today. You collect a finger stick of blood. That blood is then dropped into a tube, which is pre-filled with reagent, is mixed, and then loaded into our cartridge. The cartridge contains a QR code, which is then scanned by the instrument. And then you load the cartridge into the instrument. And after that, press start, and the entire workflow is automated to give you a result in, let's say, 15 minutes. So this is the basic, this is one of the first prototypes that we built. This is our newer version, which is kind of much more, jazzy looking basically, and a much smaller, more portable product, which is the A6 200. And that's something that, like I said, is more battery operated, smaller thing, but doing the same things, doing fluorescence detection and the microfluidic pumping with that cartridge, which is that instrument. Skip this. So what is the first product that we're doing? It's actually a thyroid panel for T3, T4, and TSH. The reason is that a lot of 8% of Indians have hypothyroidism. So it's one of the most commonly ordered tests. And many of the pharma companies that we've spoken to have said that less than 1/4 of the people that have thyroid disorders have actually been identified. So there's a need for very widespread screening to be done in doctors' offices. And it's something that you almost want to opportunistically test for, that when someone shows up in a doctor's office, you want to just say, hey, can I test you whether you have a high thyroid deficiency, right? So that is where I think point of care can really help. So for example, in a salon context, thyroid is actually linked to hair fall. And it's a sensitive subject for hair, especially for women. So one of the business contexts here is that if you could do it in a salon and you have actually a hairdresser recommend to their client that can you just get your thyroid level checked because I'm seeing your scalp, then you might actually find somebody that's just got a thyroid problem, and it can be easily fixed by taking thyroxine. So the nice thing, like I said, about the platform is you can do multiplexing. So here I'm showing you how we're doing TSH, T4, and T3, all these together on one platform, and we get good multiplex data for something like this. So that's a kind of, I would say, significant advantage of our platform over anything else that's there, that we can multiplex and do these together. We've done a lot of clinical validation where we're able to compare our platform with gold standard platforms. Let's say this is from Roche here, and show that we have excellent accuracy when compared with the gold standard platforms. And we've done a clinical trial where we took 3 of our readers and ran 300 samples to get this sort of reference data. The other thing that we're doing is actually a combination of fevers of unknown origin. Again, this is all before COVID, but one of the important set of tests to be done in India is, let's say, these dengue, typhoid, malaria, chikungunya. And these are all done today as individual tests. So one kind of common requirement, which is, doctors would really like, is can you test all of these in one shot, right? And so this is the kind of thing that we can do on our platform. So this is showing just dengue and malaria combined, right? And we did this for a large company, Becton Dickinson. So we made a product which can actually do a combination of dengue, malaria together, and kind of then working on adding the typhoid and chikungunya at this point. So these are some of the tests that are we've done and that are in the pipeline. Today, our business model is really to work on these hormone tests with large pharma companies. And we are kind of going through a clinical validation of this portable product that I talked about in preparation for a launch of that product early next or in the middle of next year to do a bunch of hormone tests in doctors' offices. The other thing is, of course, one interesting subject is, do you really need to separate plasma? One of the things that we found is for a lot of these hormone tests, we're actually able to do 2 things. One is I think I heard Amit talk a little bit earlier, you can do plasma separation, and he has some really nice technologies around this. There's also just membrane-based plasma separation, as I'm showing here, that you can integrate into the cartridge. But you can also just directly do a blood-based testing if you could measure the hematocrit. So that's something that for some tests at least works quite nicely. So you don't actually even have to do a plasma separation. You can measure directly from a blood, and we have some nice data showing that at least for something like TSH, you can directly measure the hormone from blood. So being a company, I think I just wanted to share with you a few of the learnings along the way. One is, I think it's very important early on, whatever your product profile is that you're targeting, you should actually list down in a tabular form like this, what exactly you're trying to do, right? Just so that you're clear, what is the range, the lowest to the highest limit? Are you doing it with whole blood or serum? What's the sample volume that you want to target? What's the resolution? What is the coefficient of variation? What is the accuracy? How fast do you need it? What's the cost? It might be a kind of a cliche to say, but I mean, you should put down all these things, because I know a lot of younger folks are interested in doing diagnostic testing, point of care, and so on. Important to write down a target product profile like this. That's one obvious thing. Second is, I would say, building any technology platform, there is a lot of learnings in terms of many challenges along the way. And one of the things is you need to do something that we call an error budget analysis. That simply means that ultimately a lot of platforms are judged by precision. Precision means how repeatable are your results, right? And the repeatability or lack of repeatability can come from multiple different things. It can come from operators, it can come from environment, it can come from different instruments, it can come from batch to batch variations, right? So it's just important to know what the sources of variation are. Whenever you're building a platform for scale, you need to draw up this error budget and say, okay, if I just simplified everything, I took one operator, one reader on the same day in the same environmental conditions, what's the best precision I can get? So let's say that's 4%, right? But then the moment I have two batches of sensors, that might become 7%. And then when I have three different readers, that might become 8% or 9%. So you need to do systematically calculations that allow you to do this, so that you know what all the variations that you expect are as you go along. The other thing with microfluidics is, I think the dirty secret of microfluidics is a lot of microfluidics experiments don't work, right? And everybody who does microfluidics knows this, that you only show the best results. And that's fine when you're an academic, because you're trying to really show a concept. But of course, the moment you have a product in the field, even a 2% failure rate might be unacceptable to a customer, right? If you just think about it, if you have a product and it's not working 2 out of 100 runs, that itself might get a lot of people irritated. So that's quite a high barrier, actually, if you think about microfluidic technologies. Many of them just do not have that kind of robustness. And there are many reasons why things can go wrong, but it's important to know what all those are and systematically cut them down so that you can get to a point where you're hitting the kind of robustness targets that you have. And this is a very, very important thing that I would say that anybody developing a microfluidics platform has to be honest and do, like really write down all of your failure modes. And I think there's some interesting things we've learned over the years that, for instance, you can use machine learning to remove dust, for instance, or the effect of dust. And dust is ubiquitous in India and is a big pain in microfluidic devices. But if you can find ways to eliminate dust by doing that, the simple things like that actually contribute to robustness. We actually have our own manufacturing facility, like I was telling you earlier, with a license to manufacture here. So we make our cartridges, assemble our cartridges ourselves in this our facility in Bangalore. Some of the important learnings in manufacturing, a lot of microfluidics manufacturing has multiple steps. So this is, for instance, the workflow to manufacture one cartridge. And as you can see, the bill of materials, or there might be like 15 different individual parts that go into making one cartridge. And there's multiple steps, whether they're cleaning or coating with various surfactants, assembling, laminating, packaging. All of this actually can be quite expensive. And one of the classical mistakes that people make is that they actually neglect the cost of assembly of cartridges. And you often see this with paper diagnostics, where people say, well, paper is very cheap and that, you add up everything and it's hardly a few rupees, right? But to come up with a custom manufacturing process that can actually make those things at scale might add significant costs. And that's something that you should do carefully when you're trying to calculate what the actual cost of your disposable can be. Not to mention the standard failures and so on you'll see during assembly processes. The other thing I would say is just in terms of technologies, I think for microfluidic technology specifically, it's obviously important to be aware of all the competition around. So you do have the automated analyzers like I talked about earlier, but you now also have a lot of lateral flow technologies which are becoming quantifiable. So that's what I call lateral flow 2.0. And you need to make sure that you have enough of an advantage over those technologies in order to be able to compete in the marketplace. So here I've just made a list of things, different parameters basically, and where does microfluidic technologies, where do they stand versus, let's say, lateral flow 2.0? Because this is the reality in the Indian market today. There are many products, especially coming from Chinese and Korean companies, that provide quantitative hormone analysis, right? And there are certain advantages to these, but I do believe still that microfluidic technologies can offer significant advantages. So wherever it's green are things where microfluidics, I would say, is already better, but orange and red are things that microfluidics needs to kind of demonstrate really how they are better than the lateral flow. So just to kind of go over one or two of these, I think cost is an obvious one. I think today the cost of lateral flow is very cheap. It's really a few cents or a few rupees, right? So to make a custom cartridge, oftentimes with a process that's not very mature, you can't hit those kind of cost targets easily. Secondly, a lot of microfluidic systems are closed, so they can't be read by other systems, which could be perceived as a disadvantage. The flow, the lateral flows usually just need capillary flow. The microfluidics need some pumping. So these are some of the negatives. On the positive side, I think you can really do a lot more multiplexing and just be a lot more quantitative with better limits of detection. And practically, in my experience, the quality of data on the lateral flows is not great. If you just test many of these, you'll find that the quality and the precision is not very good. So if you're able to demonstrate that to your customers, I think that's an obvious place where you can score over them. So that's one thing. The other, just from the other learning, obviously, is I think whenever you make a new technology platform, as a scientist, you're first concerned about, does it all work? This is the first question. And investors are worried about, if you make it work and you spend all the money and time, will somebody buy it, right? But in between, there is this step of, does it work at scale? And I think that's, again, something which should be, a lot of people don't think enough about, of a new hardware technology platform. As you scale it, there may be new problems that you don't anticipate at the POC level. And this is really a challenging pain point for microfluidics in general. So something that has to be thought through carefully. I just wanted to end by talking about a couple of things we've done over the years. This is an R&D platform. I know, especially because Dr. Mandal here has worked on silk, and he's done some excellent work over the years. This is a fabric-based platform that we have done at Achira, where the idea is quite simple. You take yarn, and in fact, a lot of our work has been done with silk yarn, and the yarn is then coated with reagents, and then silk being a polypeptide, actually we found that it's quite good for, in fact, storage of reagents. And then you put it on a loom, and we do just a hand loom because that's what's available easily. And then you can make effectively fabric that can then be cut into diagnostic strips. So we've made different kinds of tests with that. We've made electrochemical sensors with that by basically coating conducting inks on yarn and then weaving those in. So this black here is actually conducting carbon ink. And then you can make glucose sensors like this. We've also made lateral flow immunoassays like this, where we've really used the advantage of weaving, that what you can do with weaving is you can change the weave along the length. So you can have all of these different segments, whether it's a sample pad or the conjugate pad or the absorption pad, can be made in one piece simply by changing the kind of weave. And I think that has a certain elegance to it, unlike nitrocellulose membrane technologies where you have to cut different pieces and put them together. So this is a lateral flow platform that we've built on fabric. Again, we've shown some very interesting data and published a lot of this, so you can look it up in the academic literature. And today, we're also one big focus area for us is molecular diagnostics because of COVID and other things. So we are working right now to convert our A6 platform into a platform for molecular diagnostics in addition to immunoassays. So we have some collaborators in Germany and at Manipal University in India. And we're working with them to do a diabetic foot ulcer detection. So you take a swab of diabetic foot ulcer and you want to identify what all species are there, and in addition, what resistance genes are carried. These kinds of tests are going to become extremely important going forward. So this is a combination of microfluidics and a microarray. So this is something which is a little bit early stage development, still a couple of years away from being a product, but I just thought I'll mention it. So that's the cartridge and the instrument that we're building for this particular product, along with our collaborators. And that's it. So I'll close by just thanking, of course, most importantly, the entire team at Achira that's worked so hard to build all of these things, and all of the people that have supported us over the years and whose names are listed on this. So I'll stop right now and take any questions. Thanks.
Thank you very much, sir, for sharing your insightful knowledge. Now this is the time for queries. One question is, you have used hydrogel for sensing in pipeline. So is it reusable?
No, see, the moment you have a sensor which is, interacting with somebody's blood sample or urine sample, that itself will prevent you from using it again, right? Because now it becomes a biomedical waste, basically.
Okay, thank you, sir. I think I'm requesting if anyone have any question, then please raise your hand or. Okay, one question is there. Regarding the fabric strip, how is the adhesion?
Okay.
The question is regarding the fabric strip, how is the adhesion?
Adhesion, is it?
Adhesion.
Adhesion to what? Has somebody typed this question? Maybe I can look at it.
Okay, so somebody just write in chat box. That I am reading just. Okay, the question is how is the addition, and the next question is, sir, please show the slides of weaving again, if possible, fabric strip.
Yeah. So this is one slide, and a lot of this is described, like I said, in the academic literature. You can look up Achira Lab on Chip. There are a couple of publications on which describe quite in detail exactly what is done. So you can look at those. But this is one image of those.
Okay, thank you very much, sir. Thank you once again.
Okay. Great. So, by the way, are either Deepankar or Dr. Biman Mandal, are they there right now?
Yeah. This is Biman. Can you hear me?
Hi, Biman. Yes. Hi, Deepankar. I would, like I said, I would have loved to meet both of you. It's unfortunate that we can't meet it.
We were listening. We are not intervening because this is a students' event.
Okay.
We thought it should be like that only. Thank you for your nice talk. I know
no problem.
Biman may say something.
Yeah, thank you so much for your wonderful talk. And surely we'll connect over phone. So I'll leave you my number, and then we will then catch up. Yeah.
Sure. Thank you both, and look forward to interacting in the future.
Yeah, thank you so much.
Okay. All right, I'm signing out then right now.
Yes, thank you. Yeah.
Okay, bye.
Bye.
Thank you, Dr. Dendukuri. So this brings us to the final session of NWNTD 2020 workshop. I request Professor Deepankar Bandyopadhyay, Head of Center for Nanotechnology, and his team, Mr. Sahil Jignani, CEO, Primary Health Tech Private Limited, and Mr. Ankit Chaudhary, CPO, Primary Health Tech Private Limited, to please proceed with the valedictory function cum panel discussion.
Thank you, Konika. So I think I am audible.
Yes, sir. You're audible.
I'm visible too, right?
Yes, sir.
Yes. So I think first we should thank all the organizers for organizing such a fantastic event. I was going through bits and pieces, more or less all the lectures. Some I missed because there was a project presentation yesterday. But I tried to attend all the presentations until or unless there is some commitment. So the final one will be on something, which I like to discuss with the participants for long about this. How do you really approach about these things? First is one vertical is about how to approach these, high-end technologies, high-end science, and then high-end translation, right? The other vertical is like, this high-end translation, right? Now, this is a very, very overwhelming and perhaps a little bit risky in terms of our thought process. The way we think, the way we are brought up, we are not, generally brought up the way it is being plattered now. And that's where I think, we have to really improvise at this stage in order to make sure that that we really catch up with the time. And then at the end of the day, we really do something very interesting in our life in the form of our careers. And this is where I think some mentorship is necessary, from the colleges, from the professors. I mean, say you expect mentorship from the professors, but then, professors are also learning here. So one must, put everything on that perspective, that it's an entirely learning process, that the world is changing very fast with this revolution of this information technology, big data analytics, data sciences, different types of ICT platform coming up. You can see how quickly we change from this offline to online mode. Now people are not really very happy with the online mode. Now people want to go back to offline mode. Again, we have to adapt to certain things. Now that's where you will see the challenges will become very, very interesting in your life if you don't interpret them to be difficult, right? And that is the major essence. I just wanted to speak for about a few minutes to all of you, whomsoever is attending, especially the students, where you see, we have to give a perspective how exactly we started nano, say in 2000 or say late 1990s, when we absolutely have no clue that what exactly is nano. And then, first we did some computational part because the experimental facilities were not available. Then we went for some experiments. When the facilities came, we came to know about that. And then we were just, settling down to that shock. Then suddenly, applications came, applications related to nanotechnology. Now the product development came. So in last 20-25 years, now people want us to commercialize the products, generate revenue and all. And it is not only to a professor or a kind of well-established a professional who is supposed to do that. Now government is asking that even young people should get into entrepreneurship at the early age in order to make sure that, there is a lot of livelihood generation. Everybody need not be highly educated. Everybody need not be. Education can go in side by side, the way you are doing the things, with your profession, education can go side by side. So slowly that is the trend perhaps emerging right now, that, you need not finish your education and then you go to profession. Rather, you educate yourself while you are in a profession, and then, you evolve together with your profession or single or multiple professions, right? And that's where a lot of adjustment is necessary from our side as a professional. Those who are doing PhD right now or masters, they are in a crossroad now, whether to go for PhD, whether to go for PhD with a translational potential or fundamental. Okay, now if I put fundamental, whether I'm going to get a job or not. Okay, then if I do translational, then I have to make a startup, or I have to really go for a kind of industry job. Then how do I survive there? Now these questions are really pertinent. But my point is that, you should not be very, very overwhelmed by these questions. But then, these questions will be there for next 20, 30 years with you. These questions will be there. As I see, for last, 20 years, as I am slowly getting comfortable with all these questions, after fighting with them. And then that is perhaps the major essence of the modern profession, what we see today. It is about adaptability, and it is about how do you adjust to those questions, and then then at the end maybe you start loving those questions, right? That's also very, very important. Today I feel really happy if somebody is challenging me, because slowly that sort of comfort level, it's like a cricket match. Like when you are in the beginning of the match, the balls are swinging and all, and then you are a little bit uncomfortable, but as you play a few balls, and then as you start hitting the ball nicely, then you are into a zone, right? So I just told Sahil and Ankit to prepare a kind of 5 to 10 slides for you to show that how exactly we could really make a journey, which I don't call right now a successful one, but then at least from a place like IIT Guwahati, when I came, there was no microfluidics lab, there was no nano lab. Okay, I mean bits and pieces, the things were there, but then there was no organized thing as such. Like the lab that I have occupied right now, that was a kind of storeroom. I could see like Pranjali, who is organizing the main show, who is the heart of this particular NWATD right now. She used to do some experiments there. UV visible spectrophotometer one was there. She was pursuing her PhD, and she was doing some experiments there. And this was more or less of a storeroom, which was first converted into a lab, micro nanomechanics lab. And then it all started there. And from there, today we are having a state of art almost nearly finished. I don't claim it is finished anytime, because until or unless it is finished, it is not finished. So every day we are making hard effort. Today only I visited 3 times, in order to make sure there is no floors. Somewhere there is no floor, somewhere there is no roof. So we have to take care of all those things. But then we are nearly finishing this building, and that's a real beautiful journey. And the icing on the cake is like we can really do some translational work. Apart from doing fundamental science. Many of you know that I generally do theoretical things, and from there setting up an experimental lab, and from the experimental lab, going for the products. Okay, it just it's adaptability, and it is about listening to your heart and loving those questions. Okay, so I request Sahil to take you to that journey, what we have achieved maybe last 3 years. Okay, and then I will again come back after Sahil finishes. Sahil, are you there?
So I'll share the presentation. So can you see the presentation? I guess.
Not yet. It's only SJ.
Okay, so I'll start without presentation. Let the presentation come. So hi, I'm Sahil from Primary Health Tech Private Limited. So we are building up a medical diagnostic device. I guess now is it visible?
Yes.
Yeah. We are building a medical diagnostic device for point of care detection for last mile population. So it all started 3 years back.
Maximize your slide. Okay. Press F5. Go to the presentation mode.
Now it is okay, sir?
Still not. Perhaps it is taking some time.
Okay. So it all started 3 years back when I visited IIT Guwahati, and I met Professor Dipankar Bandopadhyay and Arun sir. And then in Dipankar sir's lab, many experiments were going for non-communicable disease. I came from a electronics manufacturing background, but then I see a lot of things happening in non-communicable disease. And when I came in contact with Dipankar Bandopadhyay sir, and then I get to know that how severe the problem for NCD is. So the major thing is that symptoms of non-communicable diseases are not apparent. So you have heard that there are patients which are categorized in a category 1, 2, 3, 4, or stage 1 or 2, 3. So what this stage means is that all the NCDs which are related to kidney, liver, pancreas, so these organs are very critical in the sense that these organs for these type of disease, if you go to doctors early in your early when the disease is started to form, then you will be saved. So a patient going to a hospital at a stage 1, then there are high chances that with the secondary and tertiary treatment, the patient can be saved. So we are working for chronic kidney disease right now. So the data shows that 50% of the patient go for diagnosis only at a stage 5, where there is the only option is to go for a dialysis or kidney transplantation. So that puts a lot of burden on the patient's pocket. So there has to be a requirement of point of care diagnostics that can be placed at remote locations at home for regular monitoring. So you have also seen this glucometer, weighing machine, all these are the POCT example, but they have changed the life of people who are suffering from, let's suppose for diabetes, glucometer is a very important tool because you have to regularly monitor the disease. Similarly, we have gone, while starting our startup, we have gone to different hospital, PHC and CHC, and we have seen that all the medical devices were imported. All these products are expensive and no Indian company is providing these devices. Even the consumables like, if you talk about buffer like NaOH, small QS, everything has to be imported, and these companies enjoy monopoly over other players. So that puts a lot of burden on patient. So what we did, we started in 2017 with the idea that we can actually make a POCT device which can actually help people by integrating 25 tests in a single device. So we got a lot of help from IIT Guwahati ecosystem, where right from technical mentorship to using the lab space, infrastructure, everything has shaped the initial idea into a proof of concept. A proof of concept means like an idea where we have got initial results. So working for 6 to 7 months, we were able to see that the device which we have actually had a vision to develop started to build up. At this stage, we have applied for various grants. We have got some equity investment from private players. So by looking at proof of concept and prototype, they have given some investment in the company. Prototype is another interesting stage of the product where after proof of concept, there are a lot of iteration has to be done. So
Sahil,
yeah.
please change the slide. Your slide is not changing. You know, on the screen, it is still, we are on the second slide.
Okay. Now is it visible? Like if I can see.
Yes, maximize it. Maximize it.
Now now you can see the slide 3.
Yes, slide 3 is now visible. Yes.
Okay. So these
but
are
it is not in the presentation mode.
Okay, there is some issue, I guess. Still the problem exists, or it has been resolved?
You magnify a little bit then. Okay. It's okay. Go ahead. Go ahead. It will become. I don't think it will work out that way.
Okay. So
demagnify it. Demagnify it.
So these are the different stages of product development. The idea, proof of concept, prototype. So at prototype, what is the expectation? That whatever product that you have built, the repetition factor comes into picture. So if you develop the prototype, that means that when you are doing the same experiment, the result X has to be obtained every time when you are doing the testing. So here the IIT ecosystem has helped in building up this overall company. They have given the access to all the latest scientific things happening in and around the campus. So at the prototype stage, what we are we got is that our process has been set. Now we have thought of going for ethical clearance. So we have applied for different hospitals like AIIMS, GNRC, GMC, and we got the ethical approval. And in collaboration with IIT Guwahati. And after that, we have started different trials. So once you are through prototype and you are going for ethical clearance, there the engineering problems start to come. So there are different diagrams which shows the development. So when you apply for a ethical clearance, typically you are at a stage 4 to 5. And here the engineering problems start to come. So when we were developing prototypes, so different things has to be taken care. Like kinetics is one of the important thing. Like Dr. Dhananjay also spoke about that. Like the time has to be, time is a very critical parameter, that less than 15 minutes has to be the time. The other thing is the stability, which has to be taken care. So we are slowly doing all these engineering parameter improvement. Right now, we have done more than 3,000 patient samples in GNRC hospital, and we have removed interferences in many of the tests. So right now, 5 tests we have completed, and we are doing clinical validation for these 5 tests. And the other 5 tests, we are at a prototype stage. We are also initiated the discussion for certification part, and we are soon going for ICMR certification for tests that we are developing. Another important thing that has to be taken care while developing product is that what is the USP that you are actually addressing while development. So what we have thought of, like the chemistry and this process, this important process is more or less same. So what we as a company were thinking that we have to build up product USP, and we have filed more than 2 patents for that. In the next slide, I'll explain about like what is the solution that we are providing right now. So our focus is to go for a decentralized healthcare monitoring. So this is the picture. This is the stage that we are currently in. So this is the device. In the device, you have to just put a cuvette. There will be a reaction that will occur, and then it is connected to a mobile application. So all the algorithms, all the processing will happen in the mobile application. So there is a very interesting thing happens. So when we were developing this product, so these products we want to develop as a standalone device. But when we were discussing with a lot of investors and the customers, so they were always saying that digital data is a very important thing that you cannot miss out. So after that, we have thought of integrating all the this mobile application with our device, so that we can get all the digital data, so that analysis can be done. And in future, we will be saving all this data in the mobile application, so that we can actually go for a predictive health insight for patients. So because in the application, we are taking medical history from the patient, and then we have also implemented few of the algorithm, so that a personalized predictive health insight can be generated. In our device, we have a vision. So while developing a product, you have to set parameters like what you exactly want to develop. So these are the features that we have these are a single slide, but these features has been in-depth thought of, like how many tests has to be integrated. So for that, we have actually zeroed down that we will be integrating 25 tests. So these 25 tests, this data came because we have interviewed more than 50 doctors in and across Guwahati. We want to place our device just like a glucometer device, and around that they are seeing these 25 tests. So this is the features that we have defined. Similarly, we want to have a device where with a single drop we can actually do all the 25 tests using microfluidics. So again, we will be taking a lot of help from people, from the professors in IIT Guwahati to implement the microfluidics in our cartridge system. We have done few initial experiments which Ankit will speak about, but this is another thing that we are focusing. And then portability is very important in all device that we are focusing, because it has to be easy to use. Otherwise, anybody who is in the remote area will not be able to use our device. So that is another concern that we are addressing. And finally, for the POCT, the domain in which we are working, affordability is the most important parameter. So we have done many engineering iterations, and we have brought down the cost by at least 1/10 of the existing devices that are there in the market. I'll also tell you another interesting thing, that when we are building a particular biomarker for kidney, so the proof of concept we have done around a year and a half back. We were getting all the reading correct, but after that we have done at least 20,000 repetition of the same test to get the sensitivity, specificity which is similar to the lab parameter. So while developing a product, a lot of patience, lot of iteration are the most important things when you are developing a product. So in our team, we have people from various backgrounds. So I have industry experience of more than 5 years. I have co-founded an electronics manufacturing company in the past, and then I have started this company. I am passed out from IIT Guwahati in 2013 as a BTech student. In my same batch, Rahul Patel was also there, who is the CEO and co-founder of the company, who looks after operations and all the investment in the company. Ankit is looking after overall product in the company. He is also passed out from IIT Guwahati. And then we have Dr. Prashant Kalita, who is a PhD pass out from IIT Delhi. He is looking after all the chemistry and biotechnology part. We are blessed with a lot of people, and we want to thank a lot of people who have actually helped us in various steps while developing this product. The product is still in the clinical validation, but without their help, it cannot be possible. There are professors like Dipankar Bandyopadhyay, Ghosh sir, Arun sir, and then doctors like Amit Dinda, who has helped in the development of the product. We have also got various funding and grant. We have won BIG in 2019. We got private equity investment from Wilgrow. We have also won few of the international competition in Singapore. And we are right now doing clinical validation in AIMS and GNRC. Thanks a lot for listening, and I will hand over to Ankit Chaudhary, who will explain about the product journey, like how the product came into picture from initial. So Ankit, you can start.
Yes, hello. Am I audible?
Yes, but there is a echo.
Yes, sir.
It's fine now.
Okay, sir, I'm sharing the slides. Yes, sir, the slide is visible?
Yes, maximize, yeah.
Yeah.
Thank you.
Okay. Hello, everyone. I am Ankit Chaudhary, and I am currently responsible for the development of the product and also the technological aspect and the principal development of how the test and the reader technology needs to be carried out. So I am passed out from IIT Guwahati in 2019. During my third year in Department of Design, I actually came across this Theranostic devices and was very much fascinated and also interested that such small devices which sits in your palm can actually detect the biomarkers present in your body to tell that what is the current functionality and the current state of your organs. So that idea was very much fascinating, which was explained to me by Dipankar sir, and that's when I started working with Center for Nanotechnology for design and development of point of care testing devices. So initially, to understand that what we need to make for low resource settings like primary healthcare centers or community healthcare centers, where you will find that barely they are having barely they're having a microscope, properly working microscope. They will have very basic instruments like the BP machines. So how to put up a device there which can actually detect the basic parameters of your body. For example, if we consider that currently diabetes is very much prevalent in India. Now for that, they are very much vulnerable for kidney diseases, and they generally develop diabetes. They generally develop the kidney disorder from diabetes basically. So what happened is that I found out that although there were 19 tests free of cost being done in the primary healthcare centers, these were dependent on the tertiary or the secondary healthcare setups of the state. For example, the samples were collected in the from the primary healthcare centers and they were sent to the government facilities. I only found one or two, barely two urban primary healthcare centers which was able to do 17 tests, out of which around 7 to 8 tests were qualitative in nature. Others were either semi-quantitative or quantitative. So that actually opened an opportunity for such devices. And the idea was, if we can empower these primary healthcare centers with such devices, then it makes the healthcare facilities accessible to the general public. Now, in the current scenario, we don't think much when we need to have a BP check actually, because the devices are such accessible. Now, the current device which we are developing is of serum creatinine, which is very important for chronic kidney disease and also for kidney diseases related to diabetes. So I went out to these primary healthcare centers and also hospitals and a few laboratories, found out that the existing devices looks like this, which is currently in my slide, which you can see in the left side. So these are bulky and huge machines, requires infrastructure. So the idea was, how can we make a device which sits in your hand but is able to do multiple diagnostic tests. So this is a story about how can we make faster prototypes and how can we make the test repeatable and make a product. So you can see the first product was very basic. After understanding what was the principle behind UV spectrophotometer, and after actually opening up a semi-auto analyzer, it is a very interesting story that I went out to one of the companies basically who used to provide service for semi-auto analyzers. I had a talk to one of the people who were the service engineer and asked them if they can show me what is the technology behind that. So what I found out is that the technology was very simple. They were using the colorimetric techniques where you use a beam of collimated rays which is passed through the sample, and as the color changes, it absorbs that particular wavelength of light, and the sensor, which is a photodiode, can detect the amount of absorbance. Now, as soon as you correlate the amount of absorbance with the concentration, you are able to get what is the concentration of, say, for example, serum creatinine in the serum. So initially, we developed a very basic prototype, using Arduinos and all. Then we slowly we were able to develop this optical core, which was very necessary for the detection part. Initially, we are using the cuvettes, which is put inside the optical core, and very interestingly, the 3D printer became my utmost friend for this. Whatever small iterations and modifications were needed, I could rapidly do it using the 3D printer, which is present here, and also back in my in the Department of Design. So you can see that these are the initial prototypes which we made, and on the right side, these are some of the handheld devices which we were able to make using the 3D printer, and also using the through-hole PCB boards, and using the components, joining it, and just making a faster prototype for the same. Now in the second part, so this actually shows that initially we are having a very simple principle, that this is the basic idea of how the device can be made, and slowly we develop prototype for it. First we developed the electronics. First we made it up to a level of proof of concept, that yes, it is working for, say for example, the initial 100 samples in the laboratory, it is working. Then we move towards doing a more rigorous test. The question is it repeatable? The basic question which always lingers in our mind, that whatever we do, is it repeatable? Are we able to regenerate the data? So this actually takes a long time and iterations for this. So the basic process is you keep on learning and you keep on improving. You try to sit with that person. You always go. What I did is I went with those people who were doing the chemistry. I sat with them. I understood what they were doing. Sorry. And what the problem they were facing, because in point of care testing you need to make the device in 3 to 4 steps. That's it. First the sample needs to be put, then with a color change or any electrochemical signal will be generated, chemical to electrical signal conversion using some transducer. Those signals need to be further quantified. Now further, after generating the first initial prototype, we are able to see, after doing a long lab tests and all, we are able to see that yes, it is repeatable. Now it is time for making a standardized protocol, that what are the dimensions of the particular device, or what particular components which we are using. What is the principle which we were initially thinking that yes, it will work. Is it working still now, or do we need to pivot from it? So these are the some things which needs to be kept in mind, and also that we cannot go blindly. We need to actually see what is currently existing in the market. For example, while choosing our sensor for our device, which is colorimetric based device, initially we thought of, let us take any type of sensor, but then slowly we understood that what are the parameters required for choosing a particular sensor. You need to read the data sheets, you need to choose a particular wavelength, you need to see the sensitivity. All these things becomes very necessary, along with the temperature, humidity of the some of the external factors. Now these are some of the prototypes which we tried for microfluidic channels. Again, these are 3D printed. And in the next part, yes, after we were through with the initial prototypes and we achieved the basic repeatability with our CV, which is less than 15% for the lab test, we developed the prototype for clinical testing, which looks like this. So the basic aim is to develop a cartridge which is with nanoparticle, with which we can improve the sensitivity. Using microfluidics, we can integrate more number of tests. Now currently to generate proprietary platforms, and also we need to validate the reagents and chemicals. Now in the reader device part, we need to make it should be easy to use, more user friendly, it should be affordable in nature, and most importantly portable. The app and the server part is being currently at the initial stage. We are getting some data and we are successfully able to upload it in the server. And also what we are able to get the correlation between the patient's basic data, what are their comorbidities and our device reading. With that, we can give the predictive analysis. So this is basically a 5-step process which we are following. First, you connect the device with our application. Then you put 1 drop of blood in our cartridge, or currently we are using the cuvette. And in the 3rd step, you operate it using our application. And all the data gets fed into our smartphone, which is being then analyzed by our application, and it is further transferred to the cloud for the patient to get all the data and his reports. Yeah, so this is the video of our company, which explains our vision. So you can see this is how our device works. So here we are getting the data from the Thank you. Thanks a lot for patiently listening. And also, I hope that I was able to give insights for the product development part. And I would now like to hand it over to Dipankar sir.
Yes, thank you, Ankit. So I just wanted everybody to listen to all of you, because you are at the ground zero. And see, I just finished, because we are perhaps we have finished our time, and maybe we should start the valedictory session as soon as possible. Just a few words that many of you have come with lot of dreams, ambitions, okay, and don't suppress them. All the students, you should always nurture them, and you should always try to realize those dreams. Okay, there will be challenges, there will be difficulties, but at the end of the day, if you really pursue them, you will see that things will emerge for you. I mean, that is the whole point. When you attempt such things, the entire process, in the form of, say, suppose if I divide your life into 2 parts, say personal life and professional life. There are many ways to divide life, but if it is, say, suppose personal life and professional life. So in professional lives, if you take more challenges, the entire thing will become very, very interesting, and you end up doing things which you are which you can never imagine that you will be doing. Like, say, suppose in Ankit's case, I can see that he was going for a design job. So the vision that was given to him, that can he end up having a design company? I mean, say, now I can see right now he's struggling, because he left 2 jobs, which he got after his 4th year of BTech curriculum, in order to do this startup. Maybe right now he's struggling to get the revenue of this company going and all. But overall, for him, the vision is to have a company. In India, design is not given that much of importance, because we don't really see the products which are very well designed. And starting from your furnitures to computers, everything you will see, there is not much of thought process that is given, because we always try to restrict or minimize the cost. But then, that is a very important aspect. When you go to a shopping mall, when you go to a different marketplaces, one of the things, say suppose you are purchasing some ornaments, okay, some clothings, design one very important aspect there. So similarly, different types of products should have different types of designs. And this particular aspect was brought to me by Ankit, that if not only about technology, not only about deep science, not only about translation, not only about market, design is also an important parameter. So and there are very less design institutes in India. So as a person, you can see that there is if you have a lot of interest in design, drawing, all these things, so what you can do, you can end up making a university, you can end up making a college rather than becoming a professor of a college. So just final words before I hand it over to Uttam and Viman for valedictory, that please pursue your dream. Okay, take risks as much as possible. At the end of the day, I think it will be very interesting. I don't see it is risky. I don't think it is risky because there are so many opportunities. Problems are proportional to opportunities. We have so many problems, right? That means we have so many opportunities. Okay, so with this, I thank all the organizers for giving me the opportunity to speak in this occasion, and I thank all of you for your patient hearing. Thank you so much.
Thank you, Divangoda.
Thank you.
Yeah. So if they have some questions, then probably we can take up. Then we can start the function.
People generally are scared to ask me questions. Okay, so I have said I am always waiting for the questions. Anyways, if they have any question, I can address.
You are very much approachable. I don't know why they fear.
Size does matter.
No questions? Okay. So Bhimanda, you can start, and then probably I can join then.
Okay. So again, formally, we this conference or workshop is quite unique again because we are doing it online. So we do not have the so-called physical participation. So again, there was no competition for students to win prizes, unfortunately. So but again, we had wonderful time. Last 3 days in terms of getting the opportunity to listen to eminent speakers who are stalwarts in their own field. Be it the industry or the academia. And the organizers are thankful to them. I mean, Center for Nanotechnology and IIT Guwahati in general. We all are thankful to them for them to spare their valuable time and then come here and then share their experience, which is very, very important because I can still recollect my days. I mean, when I was a student, and then we used to look forward to these opportunities to even now also look forward to these opportunities to hearing people and who can share so much. I mean, within half an hour or 45 minutes or 1 hour. And that is exactly what they have done, and it is so valuable because industry experts have kind of bridged the gap, which we always see in terms of academia, and then we always see the industry being on the two sides of the river, and never to be meeting together. But again, time has come, and these are the times where, and these are the efforts, I think, and we're fortunate to contribute to that kind of an effort where we're trying to bridge the gap of academia and industry. And of course, again, I take this opportunity to all the presenters and our own students who have been dedicatedly making the videos for the lab sessions. Unfortunately, we could not do it live, or we could not allow the participants to come here and experience themselves. But whatever possible, I think we tried our best, and then there was a lot of effort that was put in. So I thank everybody who has been, behind the scenes and doing it. I hope participants had a great time experiencing this new technique, and they would now be more aware and comfortable in using them in their future endeavors, be it academia or industry-related work. Of course, the principal investigators and faculty members of Center for Nanotechnology, Dipankar to start with, HOD, who has been very forthcoming in helping and being very approachable, and as a team, handholding and allowing us to do this. Arunda, of course, being a mentor figure, and of course, we learn a lot always seeing him, and being with him. Roy sir, Ghosda, all faculty members. We are newcomers, as I was saying in the inaugural session, but never felt that we are newcomers. But no, that has been the culture of Center for Nanotechnology, and me and Uttam got this opportunity, this time to have it. And most importantly, staff members who have been playing the role of anchoring and hosting, organizing. So last 1+ month, they have been at the background and trying to make these 3 days eventful and enriching. So all thanks to everyone, Anjali, Sandhan. This team is led by them, and everyone who have been part of this team. And I think I'd like to thank being part of the team, and he has been doing most of the hard effort in terms of I was mostly taking his help and guidance all time. So thank you, Uttam, as well. And who else, Uttam? Now it's your time to
Yeah, you most of the things is basically covered. Yes, I just start with this. Everything began few months back, right? And since then, there is a lot of help from advisory committee members, particularly. I definitely take few names who gave a lot of fruitful suggestions. Start with Dipankar Da, Varunda, Professor Roy, Professor Ghosh, PK Iyer. Time to time, they gave some suggestions, and then we are so basically me and Bimanda was in the middle, but most of the things executed well by Dr. Pranjali, Sandan, Anand, Martin, Sainika, Sahil, Ankit, and many more. So but these names, I know like they intensely the work, particularly Pranjali took a lot of burden and try to resolve everything because this is kind of the platform what pretty much new for all of us. So we don't really know like what's coming, right? Even though she planned really well, it's some of the point in the last moment, some of the things change, but yes, he executed things pretty well. I thank all the organizing committee members and then the students. So one of the key component of this workshop was this lab demonstrations, where they took all these burdens that have. So basically, when you see this in the finalized few hours video, right? But to record that and to finalize that, many students gave a lot of effort, a lot of hour, a lot of time, and then then only it came up like that way. So I thank all the students and also all the anchors. It was beautifully coordinated. It was amazing the way it set it up. I really fortunate that I got such a energetic and enthusiastic the group, the working group. And top of that, I again thank Dipankar and Orunda for giving us this responsibility to execute, and all the speakers who really gave wonderful talk, and like from last 3 days, different topics. I also try to listen or cover many of the talk, but this is kind of end of the semester, so there are a lot of things going on in the department and also institute level. So yeah, time to time different meetings and also, but yeah, whenever I got time, I listened them, and it was really good. Last few days, you come across with very enthusiastic talk and come up with some ideas and all. And again, this particular platform kind of encourage this collaboration and give kind of the example that you can, if you do the hard work, if you are chasing them a dream, you can really take something forward from the lab to commercialized product, which is, I think, many of the scientists have a dream to materialize something from their lab to commercialization. So yeah, so many examples are there. There are 2 panel discussions which are really amazing, give a lot of courage and enthusiasm. So yeah, with that, I thank again everyone. Yes, it's difficult to take each and individual's name, but don't feel bad, but everyone matters, and that's the reason today I think we have finished this thing quite well. Yep. Thank you.
Maybe Arunda speak a few words. SSG is there. Siddhartha.
Yes.
Please.
Hello. Can you hear me?
Yes. You are in the tea shop. We can hear from you.
I am not. I am far from tea shop. Actually, I am not reached there yet. I am not that. So first of all, I thank everybody involved. And I heard the story. This is a success that belongs to all of you. And I am very now delighted to learn that we are doing well with young people in our campus, in our building, in our center. There is nothing better than young people enjoying and prospering better than the seniors. And this gives the best feedback possible. And I am also happy that everybody involved enjoyed this conference. I watched bits and pieces, not continuously, but we hope that under the leadership of our head and very many young people, we will have many other such sessions every year. And I am hoping that that will come to be true. And we hope that next year, if nothing else, 2021, what is the program? Uttam, do you know, remember that?
No, what? No, I missed that.
ICANN. We should have ICANN 2021. That
yeah, Professor,
you can announce that we will have that, and we'll meet everybody there in person. We look forward to meeting everybody there, and I thank everybody once again. And hope you have had a great time, and you will in future we will meet and see the facility and utilize the facility. Thank you again.
Uttam, yes. We should also request some of the participants to speak up, okay?
Okay. Yeah. So, Professor PKIR, if you are around, then if you can share some thought with us. I think he's not here.
He's not there, perhaps.
He's not there.
Some of the students, some of the participants to speak up, okay?
Yeah, Rohit Kumar Kasera, if you want to share something, some thought about this conference. You unmute first. Okay. Mr. Bimal Kumar Singh Yadav, are you around?
Hello.
Yeah. Yes. If you can share your thought about this conference. Few words.
Hello. Can you hear me, sir?
Yes. Yes.
Yes, sir. Actually, first of all, thanks a lot for so many lectures that we got to hear. It was from both the academia and industry, and actually we are quite happy to leverage a lot of experimental knowledge and there, and also as a part of lab session that were so informative, and every bits and pieces were described in details. So that was wonderful, and I definitely hope that this kind of workshop will be quite an encouraging platform for the new researcher and the budding researcher. So that was for me quite an good experience, sir.
Okay, thank you.
Yeah,
Mr. Hemant Kumar Verma, are you around? Okay. Nasim Ahmed, if you can share some thought.
From the participants, if you have anything to say, please unmute yourself, speak up, no issues.
Yeah. Yeah.
Your viewpoints about the workshop, did you enjoy, whatever?
Negative, positive, both.
Yeah, whatever you like to speak up. Raghavendra is there. Raghavendra Shetty, are you there? Archini. Yeah, Raghavendra.
Sir. Good afternoon, sir. Hello,
yes,
sir. Are you hearing, sir, my voice?
Yes.
Yes, sir. Sir, I would like to say, first of all, thanks to organizers and speakers for delivering good lectures. Sir, actually, I am from mechanical engineering department. I have little bit of knowledge of nanotechnology, sir. But by attending this 3 days workshop, I have got the knowledge of in deep, means micro machining. Actually, we know the macro machining, sir. But by attending this job, how we can manufacture by using nanotechnology in micro machining technology, sir. Sir, this is a thing I got in deep, sir. From my side, I would like to say thanks to everybody for organizing this wonderful workshop. Thank you, sir.
Thank you. Thank you. Tulunga Basmutri. Can you share your thought? Hari Kumar. Probably network issue or something.
Yes, maybe others are not that much. It's okay. Maybe we can continue then, Uttam. Huh?
Yeah. So, Pranjali, if you can share some of your thought about this conference.
Hello, am I audible?
Yes.
Yeah. So this time, this is the 6th national workshop we are organizing. So this is the first time we're doing the online. So we are also a little bit confused, like how it will go and how we can organize the things. So then because of the 1 to 5th workshop was offline. So we are a bit familiar with the offline procedures, but online is little bit like worrying. Me, Sandhan and Anand, we usually discuss among ourselves like how we should plan the things and all. But finally everything goes nicely. So, and thanks to all the participants, to the conveners and the faculty members. So this is what I feel. Thank you, sir.
Thank you. Sandhan.
Thank you, Pranjali. Thank you. I mean, you can see from everybody, you know, speaking out, and everything was so well organized. Everybody's praising, you know, all you and your team's effort. Thank you so much once again.
Yes, thank you, sir.
Yes, special mention about Pranjali and her team. Okay. So Pranjali, fantastic job along with Sandhan, Anand, Sahanika, okay, Indrajit, Kaustubh, Ayon, Manjay, okay, Emmeline, I can see Franco, okay, all of you, Rina, everybody participated. Okay. So that is one point, and then we also like to thank all the administration. I mean, AER office, DIN-R&D, okay, directors of everybody, like, you know, helped us like anything in setting up the things, giving us the boardroom, okay, the conference rooms, everything where whenever we asked, whatever we asked, it was supported by the administration. So we also like to thank them.
Right. True. Yeah. So with that, we'll conclude this conference here. And thank you so much, everyone, again. Thank you, Varunda. Thank you, Dipankoda. Thank you, Bimanda, and everyone.
Yes, thank you all. Thank you, Uttam. Thank you, Biman. Thank you all for your participant, for your organization as well as okay. Thank you so much.
So thank you all. So then we can close the workshop then. Thank you. Thank you, everybody.
Bye-bye.